this afternoon's breakout session for the broiler industry. Our moderator earned his Doctor of Veterinary Medicine from the University of the Philippines, Diliman in 1988, and he specializes in poultry medicine and management. Early this year, he was awarded the Outstanding Veterinarian in Farm Animal Practice for 2021 by the Philippine Veterinary Medical Association. The Veterinary Practitioners Association of the Philippines also chose him to be the awardee for the Outstanding Poultry Practitioner in 2014. He has more than 3 decades of experience as a practicing farm veterinarian and has served in the corporate agribusiness sector, taking on technical service management and marketing positions in various multinational companies like Cobb Asia Pacific, HIFRA Philippines, and others. At present, he is a consultant and a lecturer of various local and multinational companies. Ladies and gentlemen, one of the finest consultants from PCPP, please welcome Dr. Raul Elias C. Lopez.
Magandang hapon sa inyong lahat. No? Mayong hapon sa inyong tanan. Thank you very much for attending and participating actively in the PCPP Poultry School 2021. We are very happy of your attendance. Maraming salamat po. Ngayong hapon, we will start our breakout sessions of poultry in broiler and layer. So this is the broiler session. And I'd just like to remind everybody that this afternoon we'll have 5 speakers and they'll be talking about various topics with regards to broiler production. Kaya abangan po ninyo. And don't be afraid to ask questions. Please ask questions, things that are relevant to your concerns, and you can put your questions on the chat box. No? For those that are not familiar sa Zoom system, the chat button is on the bottom part of your screen. No? And doon yung chat. So you can put in there your questions. All the questions will be more or less, or some of the questions will be posed to our speakers by the end of the presentation this afternoon. No? So abangan po ninyo lahat yun. No? For the questions, we'll try to send all the questions. If they're not going to be put forth to the speaker and answered, we will send them to the speaker and we'll ask them to reply. And the questions and answers will all be collated, and then we will put it up on the PCPP website, which is the www.pcpp.org.ph. So makikita nyo lahat yung questions natin doon and the answers of our speakers. No? Also, don't forget to evaluate the speakers at the end of the question and answer this afternoon. Very important, so we can improve even better how we conduct the PCPP Poultry School. Of course, you have to take the quiz, especially for those that are applying the points that will be given for your veterinary license. No? So right now, tayo are going to proceed now with the first speaker. No? And our first speaker, his paper will be about the advances in omics or genomics and its relevance for poultry production and gut health. Okay, our first speaker this afternoon earned her degree as engineer in food science. She also got her master's degree in production management, logistics and purchasing, and also her doctorate degree in biotechnologies from the same university, which is the University of Caen in Normandy in France. She served as a senior scientist at Sorbial SAS from 1998 to 2009, and currently she is an associate professor at the University of Caen, Normandy, which she had been a professor since 2008. So currently, she is the principal scientist at IFF Danisco Animal Nutrition. So ladies and gentlemen, please welcome Dr. Marion Bernardo.
Hello, everyone.
Good afternoon,
My name
Maria.
is Marion Bernardo. I'm working for IFF Animal Nutrition as a project manager, and I'm also an associate professor at the University of Caen Normandy in France. It's an honor for me to participate to this poultry school. I would like to thank the organizing committee for inviting me to talk about advances in omics and relevance in poultry production and gut health. As an introduction, it's good to set the scene and remind everyone that global demand for meat has increased more and more over the last decades, driven by the growth of the population, the increase of the incomes, and urbanization. Within the livestock sector, poultry emerged as the most efficient subsector in its use of natural resources and providing protein to supply this global growing demand. Poultry birds have a particular role to play in improving nutrition in developing countries for providing affordable, high-quality protein and essential nutrients, without being associated with any of major religious and cultural taboo. This explains the 5% average annual growth rate for poultry meat over the last 15 years, while it was only 1% for meat, 1.7% for small ruminants, and 3.1% for pork. The global production for poultry meat for 2021 is planned to reach 102 million tons, mainly thanks to the advance in breedings, zootechnics, nutrition, veterinary medicines to improve production rate. However, in today's poultry environment, where there is an increased pressure to produce even more while reducing antibiotics use, there is a need for a major breakthrough to control pathogen and gut health that inevitably become essential. The good news is that the omics technology appeared as a real game changer. First developed and applied in humans at the beginning of the 21st century, omics refers to collective and high throughput analysis, including genomics, transcriptomic, proteomic, and metabolomics that integrate through robust systems, biology, bioinformatics, and computational tools to study the mechanism, interaction, and function of cells, populations, organs, and tissue, and the whole organisms at the molecular level in a non-targeted and non-biased manner. For the last decade, it has been integrated into animal sciences, and the aim of this presentation is to give an update on the latest advances and values of omics technologies applied to broiler chicken. Genomics is the first omic technology that has been developed. The last version of the chicken genome sequence released in 2017 is named Gallus 5.0. Compared to the previous genome, the previous version, it contains new sequences, showed a tenfold increase in content length, and added 4,700 annotated genes, both protein coding and non-coding transcript. It provides a substantially improved gene set for the future research. As genome sequencing becomes easier nowadays, genome annotation remains highly challenging, while with most of existing genome annotations being incomplete or prone to bias and errors. Major efforts are now focusing on appropriate functional annotation. It's important to collect genomic information from large population of animals to discover, preserve, and utilize this genomic diversity. Genomic can also support the development of high throughput systems and will help to understand immunity of avian diseases, which is not fully understood. Thus, highly annotated genomes can improve poultry production. This is exactly the goal of the Functional Annotation Animal Genomes, the FAN consortium that was created in 2015. The FAN consortium is working to discover basic functional knowledge of genome function to decipher the genotype to phenotype link in farmed animal. We call it G2P. The achievements of all the found research priorities will together increase the capacity of the farm animal production industry to face the challenges of the future, empowering genomic selection, enhancing adaptation to changing environment, conserving biodiversity, and bridging the gaps between cellular and all animal scale knowledge. We're all looking forward to see the outcomes and poultry production applications of this group. Chicken genomic is thus crucial, but more recently, scientific attention focused on a virtual organ or system with functions of importance to the host. In the last 10 years, it has become increasingly apparent that the gut microbiota plays a significant role in host biology. This virtual organ has been studied for ages, but the level of knowledge has increased significantly since omics technology have been available and applied. The first description of the chicken gut microbiota dates back in the 17th, but was limited to a number of bacteria genera, basically the one that we were able to grow in the labs. More in-depth knowledge came in at the beginning of this century with molecular technique for rapid fingerprint analysis of microbial community composition, like the DGGE and TRFLP. But the real breakthrough came in the last 5 years with metagenomics applied to the chicken microbiome. We then discover its unsuspected diversity and richness, opening the door to a new exciting and promising research area, as illustrated by the growing interest of scientific groups in chicken microbiome, which has grown by a factor of 10 between 2015 and 2020. Those culture-independent approaches gave new insights of gut microbial diversity, and especially its dynamics across the GI tract. Around 700 phenotypes are now described, and a lot of new species have been identified. The dynamics of the microbial population is also a new element with significant differences in the establishment according to gut location and a long time, as illustrated here by the work from Oakley and Kogut in 2016, which has been confirmed in a consistent manner recently with the work from the group of the University of Liverpool in 2020. This is an illustration adapted from the work of Shelfie published in 2015 of the differences in the gut microbial population according to location and its dynamics along animal development. Besides proteobacteria and firmicutes, lower abundant phyla change significantly with time, indicating high dynamics in the reorganization of the whole microbials through time. It's important to highlight that at the early stage, scientists noticed important individual gut microbial variation, as shown by Sargent in 2014 in this graph. It shows the proportion of the top 5 most abundant OTUs in the cecum of broilers at 42 days of age, but from the same commercial flock. As you can see, the proportion of bacterial groups differs significantly in abundance and diversity. This already suggests difficulties to define what is the healthy gut microbiota composition of a broiler chicken. Despite these new findings and recent publication, it's still difficult to assess what is the composition of an healthy chicken. This could be linked to several factors. The limited numbers of publications showing also controversial results, the differences in the experimental methodologies that have been applied, or the rearing conditions that have been used. Just to illustrate, here are examples of contradictory results related to the stabilization of the gastrointestinal tract microbiota. According to Apalagi, Liu, and Kong, respectively in 2004, 2003, or 2008, the gut microbiota is early stable after 3 days. For Shofi and Awad in 2015 and 2016, it's not stable until day 14. For the others, fluctuation still happens after 11 days or 25 days. In the review published in 2020 on the composition and function of chicken gut microbiota, Reichlich said that the core gut microbiota of an adult chicken cannot be defined before 20 weeks of age when it's stabilized. Thus, in the broiler chicken production system, we are working with juveniles exhibiting a non-stabilized gut microflora that is influenced by many environmental factors, which makes it difficult to determine. Another aspect that makes it difficult to make any general statements about the absolute bacterial density in the different parts of the chicken intestine is because it is difficult to compare one study to another, either because experimental conditions are not always described in details, or because parameters are not the same, making meta-analysis more difficult to run and interpret. Oakley in 2014 compiled the data of phyla composition of chicken Sika made by different scientific groups, and he noticed differences in nature and proportion. By looking carefully at the experimental protocols, he noticed either the lack of information or differences in the type of breeds, the source in the gene bank sequences, the type of diet, etc. And we know how important the type of diet is in the composition of the gut microbiota. As an illustration, just looking at the color code, it's obvious to see the different microbial patterns of birds fed a corn-based diet or wheat-based diet. In the recent article, there is another example that reveals that not only macro ingredients, but also micro ingredients like calcium, phosphorus, or additive as fructooligosaccharides affect the composition of the ileal microbiota. So as to conclude on this experimental part, the methods applied over decades are not comparable, and comparison between studies is not meaningful and explains why it is still difficult to establish solid trends on what an healthy gut microbiota is. Thanks to the functional gene-targeted metagenomics, it was also discovered that the gut microbiome is not a silent or a freeloader. Besides digestive functionalities, like the synthesis of vitamins, short-chain fatty acids, gut nutrition and digestion, the gut microflora also plays an important role in non-digestive functions. We can cite the competition with pathogen, maturation of the epithelial barrier, training and regulation of the immune system, and much more are about to be discovered. As we discovered the scope of the gut microbial role and potential, it became crucial to have a comprehensive analysis and understanding of the interaction with the host and environment to further implement strategy to improve gut health. Thus, beyond mapping who is present in the gut, technologies were developed to better understand what they can do and how their metabolites interact with the host according to environmental factors as the age, the genetics, the medication, the production practice, and so on. This slide illustrates part of the array of the omics disciplines that have emerged. From chicken intestine samples, we can now determine which bacterial population is present and relative abundance of the different groups. But basically, these bacteria can be considered as cell factories. So more than knowing about their presence, it is important to understand their functionality potential, what they can do, and what they are being done in certain conditions. So more interestingly, thanks to the transcriptomic, it is possible to reflect active genes that are expressed at a given time. With the proteomic, we can define the entire set of proteins that have been produced or modified. And finally, with the metabolomic, it gives us the opportunity to study a complete set of small molecules that we can find within a biological sample. Thus, it's possible to modify the environment, the diet, the production practice, and see how it affects the diversity, the abundance, the richness of the intestinal microbial population, directly impacting the secretum and the host response. And surprisingly, it was noticed that despite chicken can exhibit taxonomically variable community composition, the functionalities tends to be conserved. Just to illustrate the value of getting the full picture and not only focusing on the microbial population. I will now use the rest of my presentation time to illustrate with examples taken from the scientific literature how the understanding of the composition and the function of the chicken gut microbiota may allow for the construction of new accurate nutritional strategies and thus improve the gut health and the pathogen control, reduce the need of the antibiotic therapy, and improve chicken welfare in the broadest possible terms. The first example illustrates the interplay between the commensal microbiota and the chicken immune system and function. In this study, authors established the correlation between gastrointestinal bacterial community by sample type, fecal and cecal, by time, 1, 3, and 6 weeks after hatch, and they measure the mRNA transcription of pro and anti-inflammatory cytokines. Several Firmicutes genera, such as Caloromater, were negatively correlated with pro-inflammatory and positively correlated with anti-inflammatory cytokine expression. It's consistent with the growing body of evidence demonstrating positive influence of Firmicutes on the gut health. On the opposite, transcription of pro-inflammatory cytokines were positively correlated with proteobacteria. Although the reasons and precise mechanisms of the alteration of cytokines induced by the intestinal microbiota are not fully understood, the trend of studies to date indicates that an immunomodulatory strategy by controlling the gut microbiota at the early time point may be beneficial. I included here 2 recent references of in novo injection of dietary supplement, prebiotic and probiotics, which positively modulate molecular response in intestinal and immune tissues. This study aims to assess the effect of the inoculation of lactobacillus species immediately post-hatch on cecal and ileomucosal microbial community. I selected this study because we are given the host response ahead of the microbial data, which is not always the case. Here, we already know that birds inoculated with the probiotic mix reach significantly higher weight by 20 days of age. Then, it's interesting to figure out the effect on the gut microbiota of the supplementation. Authors demonstrated that probiotic strain from the mix do not have the same colonization profile in the gut, one strain being more persistent, which is Lactobacillus inglovii. They also showed that the probiotic blends modify the microbiota development and structure with a tendency of promoting beneficial taxa while reducing pathogenic ones. It is then possible to modulate and orientate the early establishment of the gut microflora by the supplementation of a specific probiotic strain in a way to promote weight gain. In this study, a challenge with Clostridium perfringens was induced, and authors assessed the benefit of diet supplementation with essential oils, another feed additive. As for the previous study, it's important to demonstrate the evidence of a macro effect on the animal. Here, the significant reduction of the mortality and the reduction of the lesion score of the challenge supplemented birds compared to the control. Then, trying to figure out the mode of action, authors compared the relative abundance of bacterial groups and showed that the abundance of some specific species of the lactobacillus group, like Crispatus or Agilis, were increased, as well as E. coli. when they received the essential oils, while other species of lactobacillus were decreased. That was the case for Lactobacillus salivarius and Johnsoni. They also looked at the gene expression of virulence factors and showed that the combination of thymol and carvacrol supplementation reduced the abundance of genes encoding factors related to ATP-dependent protease, endotoxins, and bile salt hydrolase when they compared to the non-supplemented control. Thus, the reduction in gut lesions could be due to the essential oil's effect on mediating the pathogenicity of Clostridium perfringens in the gut, reducing virulence factors. This is another example combining metagenomic and metabolomic. Here, authors compared the effect of antibiotic or probiotic supplementation on the gut microbiota composition and abundance, as well as the associated metabolome. Data are not shown here, but both supplementations significantly improve the body weight gain and the average daily gain compared to the control birds. We do see on the graph on the left that the cecal microbiota is modified by the supplementations. The probiotic supplementation significantly increased the relative abundance of Faecalibacterium on day 21 and 42, and decreased the Ruminococcus on day 42. The associated metabolome showed that dietary Bacillus amyloliquefacients changed the cecal metabolites involved of amino acid metabolism and glyceride metabolism, while the antibiotics changed the cecal metabolites that were related to the carbohydrates metabolism and also amino acids. So this study illustrates that probiotic can substitute antibiotics growth promoter in improving growth performances, but the mode of action is different, as we can see by the associated secretome. Another example, slightly different. In this study, authors are interested in the genetic selection of digestive efficiency, a component of feed efficiency. The aims were first to identify predictive biomarkers of apparent metabolism energy using the serum metabolome, and to unravel the metabolic pathways influenced by digestive efficiency by analyzing the covariation of digestive efficiency trait and the metabolome from ileum and cecum content. In their tested conditions, metabolites presenting the strongest associations with the apparent metabolism energy were proline in the serum, fumarate in the ileum, and glucose in the ceca. This study showed that the serum metabolomic offers new opportunities to predict chicken digestive efficiency, and it may help in the development of more sustainable poultry production schema by allowing a better combined nutritional and genetic approach to production in a context of a more variable and complex composition of broader diets. Maybe less familiar is the term of immunomics. It's another example of the array of omics technologies to provide insight as to how immunity-related genes are expressed with regard to different situations. This is highly relevant to improve the fundamental knowledge of the maturation of the immune system and the role of the gut microbiota in healthy birds, as well as during disease progression. It is worth mentioning that recently, the immune genes of more than 300 bird genomes were released in a public site called the Avian Immunome, which will help us to better understand the immune system, which is quite different from that of mammals and still not fully understood. This example relates to Avibacterium paragarillarum, the causative agent of infection coryza. This study aims to look at expression patterns of chickens infected with this bacteria. Authors have identified the toll receptor signaling pathway as being significantly enriched in infected birds versus control. And the immunomic tool enabled to identify the entire cascade of the immune response from the initiation to the production of cytokines. And this knowledge is important since the exact immune defense mechanism is poorly understood for this infection. And by generating fundamental information, by identifying genes that might encode for protein that can act as an answer for immune function, will serve the vaccine strategy. Another example that is based on the identification of different immunometabolic signatures identified in the cecum chicken infected with Salmonella enteritidis. This study has used the emergent field of kinomics, measuring the immunometabolism of the tissue level to identify potential mechanisms by which the host can tolerate Salmonella infection. The chicken-specific kinome array reveals that Salmonella enteritidis modulates host immune signaling pathway in the cecum to establish persistent infection. The pro-inflammatory response identified at the early stage of contamination is followed by a sort of tolerance status between day 4 and 14 post-infection, before the animal reach a third stage of non-inflammatory status, which we can call the homeostasis. Taken together, identifying potential molecular mechanisms of disease tolerance of a host defense can not only provide a perspective into the evolutionary forces that have driven the coevolution of the host with the pathogen, it also provides the discovery of new therapeutic targets to control foodborne pathogens. Not only infection modulates the immune system, feed ingredients can also interact with the functioning of the immune system. Here is a study where authors looked at the kinome and biological immune response of birds fed a diet containing additional beta-galactomannan with or without mayonnaise. The inclusion of additional beta-galactomannan into the diet resulted in an increased signaling related to immune response compared to the normal control diet. But the addition of the beta-mayonnaise in the diet eliminated the majority of this immune-related signaling, as seen in the changes in specific metabolic and gut function pathway. These observations are likely the mechanisms for the enhanced performance and the feed conversion observed in the birds given beta-mayonnaise in their diets. I will stop here the examples, despite much more exist. And this highlights the real breakthrough of the omics technology. But there are still challenges that science needs to overcome. The limitation are the need for the improvement of the macro and the microorganisms' genomes, the cost of the DNA and RNA sequencing and the mass spectrometry, the need for computational capacities, the need for uninterrupted development of analytical tools to analyze the huge amount of data that are generated, and the need for the integration of the data. It is time for me to conclude this overview. I hope you agree with me that the application of this cutting-edge omics technology paves the possibility to understand much useful information about the biological processes and pathway behind different complex traits of chicken, with important knowledge achieved in the field of feed efficiency, nutrition, meat quality, and disease resistance in broiler chickens. But as we know more about the phenotypes of these complex organisms that is shaped by the bidirectional interaction between host organisms and its associated microbiota, it becomes obvious that exploring the full extent of these interactions requires carefully consideration of study design for the efficient generation and optimal integration of data derived from the metagenomic, metatranscriptomic, metaproteomic, and metabolomic from both hosts and microbes. In this perspective, the holo-omic approach was introduced 2 years ago and tackle the interplay between the two. It is covered by two projects. One is ongoing, it's called Holo Food, and more specifically, Holo Chicken. It explores the interaction and impact of the microbiome together with the host chicken in response to different feeding strategies that are already implemented commercially. This project started in 2019 and is supposed to be completed in 2022. The other European project just started the 1st of September 2021. It's called the 3D Omics, and this one is expected to bridge the technological gaps that I listed previously. It aims to develop, optimize, and implement the 3D omic landscape in animal production to generate the most accurate reconstruction of intestinal host microbiota ecosystem ever achieved. 2 terrestrial production systems have been identified as priority, locally poultry and swine. A myriad of factors, including animal development, diet, exposure to pathogens, and management practice will be integrated. Deliverables for this project are expected to bring production animal to a new frontier. Then we have a date to meet in 2025. And having said that, thank you for your attention, and I'm now ready to take questions.
Thank you, Dr. Marian. Excellent paper. Very, so many details, and I think that's the future. We have to understand what we have with regards to organisms that we have in the gut, especially for broiler production. So thank you very much. Our next speaker will be presenting about reducing antibiotics in poultry productions, competitive exclusion, and innovative approach. Our speaker earned her degree as engineer in animal health from ENSAR, which is the École Nationale Supérieure Agronomique de Rennes in Rennes, France. She got her doctorate in poultry physiology of reproduction from Tours University, INRA, the French National Institute for Agricultural Research. From 2011 to 2015, she worked as a poultry nutritionist at Envisions Turkish Limited, UK. She was a poultry scientist at Tecna, poultry specialist at Tecna, a premix company in France from 2015 to 2016. And in 2016 to 2019, she held the position of technical manager for egg-laying hens at the French feed company Sanders. A poultry consultant in 2019, and in 2020, she took the position of product manager and poultry technical deployer at Lallemand Animal Nutrition. Ladies and gentlemen, let's welcome Dr. Corinne Morvan.
Good day, everyone. My name is Corinne Morvan. Thank you for the invitation to this Poultry School Online. I'm really pleased to have this talk today with you, talking about reducing antibiotics in poultry productions. I am working for Lallemand company. I joined the company last October, and I used to be working for the poultry industry in different countries for 20 years as a feed manager or a poultry specialist. I used to work for feed mills, for premix companies, and for a genetic company, which is Envisions. And I did some field support and also some nutrition support since I used to work as a nutritionist. So my talk of today is to explain you why a competitive exclusion product is an innovative tool that you may use in your process for reducing antibiotics usage in poultry productions. As an introduction to my talk today, I would like to focus on the importance of the poultry productions. And we know that this poultry sector is benefiting from the most significant growth to cover the protein demands worldwide, especially in your area in Asia. By 2030, we expect a growth of more than 16% for just for the poultry meat, and talking about egg production, by 2025, a growth of more than 7% is expected. So this is really the market to work in, and this is where we will see the more profitability in the animal sector. The poultry industry is one of the largest contributors to food safety, unfortunately, especially talking about Campylobacter contamination, which is the first contamination, the first bacteria which is causing contaminations in poultry products, but also Salmonella, which is a high and very huge concern for countries' authorities, health authorities, just to manage human health and food safety. And poultry operations are very sensitive to environmental challenges, and antibiotics are widely used to treat disease. And in some places, it is very cheap and very easy to use antibiotics. But we know it's not the right solution in terms of food safety and human health, because of antibiotic and antimicrobial resistance that are emerging in those poultry products. So it is really crucial to change our process to reduce antibiotics usage. And managing gut health is really important and critical for birds' performance and for health. So this is the agenda of my talk today. In the first part, I would like to present you the gastrointestinal tract of the bird. Then I will explain you why the microbiota is so important to support the gut health. And then I will come to the competitive exclusion principle and explain you in details why it is an innovative tool to reduce antibiotics use. So the first part, description of the gastrointestinal tract of the bird, which is important to understand how it works and why the microbiota is so important. So there in this slide, you have a description of the digestive organs in poultry. So you may know it already. I just make a short description and reading of the different organs involved in nutrition and digestion in poultry. So you have the crop there, which is this part. Then just right after the proventriculus and the gizzard. This first part is related, those first organs are related to what we call mechanistic digestions. This is where the feed is going to be grinded into small particles that will allow a better digestion in the rest of the digestive tract. So if we go on with the description, here you have the pancreas. You can't really see it on the picture, but it is really between those segments of what is called the duodenum, the proximal part of the intestine, the small intestine. There you have the jejunum with the Merkel diverticle, which is an indicator of where is the jejunum, and the last part of the small intestine, which is the ileum. This is where the enzymatic digestion is going to happen, starting with more with the mineral absorptions, calcium and other minerals like sodium, phosphorus, etc. It is a continued process for digestion. There, if we look at the pattern of the epithelial cells, we have some very long and developed villi, because this is where the digestion is starting. And the particles are a little bit bigger. In the jejunum, the villi are a little bit shorter, and this is where mainly fat and starch and protein and also the rest of minerals are absorbed. And in the last part, in the ileum, it is just the end of the digestions for those same metabolites. So in this small intestine, the digester are emulsified with some bile salts and enzymes, and this is where we have the main nutrient absorptions in poultry. And the last part is where the fermentative process happens. This is what we call the hindguts, and you have two parts. You have the last one, the colon, which is just before the cloaca, and those two parts there, which are the ceca, so one cecum and two ceca. There it is really the last part of the nutrients absorption whenever it is possible, and when the fermentation process is not too much, is not taking when the balance between fermentation and nutrients absorption is correct. So this is where you will have the digestions of celluloses, polysaccharides, also some short-chain fatty acids absorption are going to happen there. And the cigar are also very, very important for water absorption. This is why in some cases, when you have some dysbacteriosis, and you can see it in the field, when you have some poor feces quality, the birds are drinking a lot. And one of the explanations is that because the cigar are not playing their role for a good water absorption, so the birds need to drink more. The whole process of nutrient absorption is very, very short in poultry, only 4 to 6 hours compared to mammals, for example. So it is a very quick process, and you need to make sure that all the nutrients in your feed is going to be completely accurate for the birds to have the optimal absorption and take the most of the feed you are going to give to the animal for optimal performance. The development of the gut at least starts before hatch, during embryogenesis already. And there you have some pictures showing the development of the intestine, already 18 days of incubation. So 3 days before hatch. This picture has been taken between transfer from the incubator to hatches. And there you have a picture of a bird at the day of hatch, and there an histological observation of the intestine. So the forms there are what we call the villi, and you see that at hatch, so it is already starting to develop before hatch, and it is not completed at the day of hatch. And you see that you have an increasing number of those dark points. Those dark points are the goblet cells, the ones that are producing the mucus, and the mucus is going to make the protection layer here above the villi, and it is going to create a barrier to protect the intestine from all the poor detrimental bacteria that are going to develop here in what we call the lumen, so inside the gut. So the incubation practice you will engage at the hatchery level is very crucial for the development of the bird, but also for the gut development. And the microvilli are not completely formed at hatch. And now I'm going to show you that the process is still continuing after hatch. It takes around 2 weeks for the gut to reach its final size and functionality after the startup period. So here you have an illustration. It's quite an old data, but still reliable and representative of what happens. Here you have the development of the villus volume and the number of enterocytes, so the cells of the epithelium of the intestine. And if you remember the 3 parts of the intestine, so the first one is the duodenum, the second one is the jejunum, and the third one the ileum. So you can see that the first one to develop is the duodenum, and there you see the multiplication, the very high speed of multiplication of the number of cells in the duodenum, and then it closes a little bit, so you see that this volume of the villi is not increasing anymore, but it takes more than 5 days to reach the volume for the birds to start and have a good feed and nutrient absorption. And then you have the jejunum, and the last one to develop is the ileum. So it's very important that to understand that it takes 10 to 2 weeks of age for the birds to have a complete development of its digestive tract. And if we look also at the enzymatic functions that I mentioned in the first slide of the description of the digestive system in poultry, you can see also that it takes 4 to 10 days for the bird to establish the main enzymatic activities for an adequate nutrients absorption, like protein. This is what we see with the trypsin enzyme, and you see that the trypsin release in the blood is increasing still increasing at 10 days of age. The same for amylase. So the level is really lower, but it is still increasing. So this is what is showing how the capacity of the bird to digest carbohydrates. And lipase also increasing during the 4 or 5 days of life, the first 4 or 5 days of life of the poult, and this is showing the capacity of the bird to digest fat. And what we can see is that during the first days of age, it's easy for the bird, quite easy, because the level of trypsin enzymes is quite high compared to the other enzymes if you compare the levels in the scale. So it also shows that birds can ingest protein and use protein for their growth, and they need it for the cells multiplication, for the body size, but also for the gut development. It's very important. And you can also see that it's quite hard for the chick to digest carbohydrates and fat because the enzymes is still not there and the level is quite low for the bird. So it's important to understand this process when you need to take care of chicks that have just been placed in a house. And this explains why you need to put some special care to those young chicks. Here, I want to insist on the importance for the chick to have access to early feed, because this is the first feed intake that will trigger the yolk sac absorption. We have in mind that if the birds don't have access right away to the feed, it's not important because there is a yolk and the chick is going to use it. But in fact, this is not true. It is completely the contrary that is happening. The birds need to have access to feed to be able to use the yolk. And this is what the study is showing. There you have 3 lines. The blue one, at least the low one there, is where birds had access right away after hatch to feed. This one is where the birds had access only 1 day after hatch to feed, and this one 2 days after hatch to feed. So you see the line, the weight of the yolk is decreasing, which is normal because the bird is using it. But the birds is going to use the yolk as quick as it has access to the feed. So the best use of the yolk is when birds have access right away to the feed. And you see that the decrease is lower when the birds don't have access to the feed. So this is very important. The feed is going to be motivation. It is going to be the starting point for all the process to be engaged. And it is also very important for the complementation of the gut development. Here, just another photography to illustrate this effect of feed deprivation on the gut development. You have in this first organ or first digestive tract, you see that the birds were fed from the beginning and it is a normal digestive tract. We still have the yolk there. Those organs have been collected at 2 days of age. After 8 hours of feed deprivation, you see already, at least the patterns are correct, but we can clearly see that there is no feed inside and it is a little bit shorter. So we can clearly imagine that it will soon be a problem for the chick's development later. And after 2 days of feed deprivation, the yolk is still there. So you can see that the chick hasn't been using the yolk and there is nearly nothing. You see that it is the intestine is very fine, and I can tell you it's very smooth. There is no it's not hard, it's not working well. And in the cigar there, you have nearly nothing and maybe some blood even there because there is no feed in the intestine. Another very interesting study, which is more recent from 2020, is making the link with feed deprivation and how the microbiota may work and play its role within the lumen and protecting the epithelial cells, because there the authors took some pictures from the intestine. So this is the intestine, the epithelium cells, and here you have the villi. So the first picture, this is at the ileum level. So The last one, the last part of the small intestine. Here it is a non-deprivated group. Here the birds had access only 24 hours after hatch to the feed, and there 36 hours to the feed. And what we see again there, the black points are representing the goblet cells producing mucus. And you see that the more the birds have been deprivated from their access to the feed, the less goblet cells you may see here on the picture. It means it is completely modulating the capacity of the birds to produce some mucus, which is going to make a barrier and the first gut protection against pathogens. So, and we know that the first hours and the first days are really critical for the birds if we want to make sure that the birds are not going to be colonized by any pathogens like Salmonella. So let's switch now to the importance of the microbiota to support gut health. There you have a picture. Don't try to read everything. Just look at the colors and understand the differences there are between the different parts of the digestive organs. So here we start from the crop, and we are going along the digestive tract until the final part, which is the ceca and the colon. And clearly what you can see is that the bacteria microbiota profile is quite simple at the beginning in the first part of the digestive tract. And when we are going to the ceca and the colon, and especially in the ceca where it is a fermentation process, you can see that there is a much higher diversity of bacteria content in the ceca, which is completely normal. And what we are measuring when there are any dysbacteriosis or immunodepression or inflammation is how this profile is moving from one balance to another. So it means that between two groups of birds or two birds with one in good health and one in poor health, you may have some different profile in bacteria. And we can see also that there are some major bacteria like lactobacillus. We also have some Bacteroides, etc. So there are some genera that we find very regularly in birds. So this picture was taken in quite a young age, and we know also that it takes nearly 2 to 3 weeks for the birds to establish its own microbiota, especially because after 2 weeks of age, the bird is able to produce B lymphocytes, which are going also to modulate the balance of the gut microbiota. And the microbiota functions have been described already a lot. There are plenty of articles which are proving, making evidence of the role of the microbiota. You may know already that 70%, more than 70% of the chicken immunity is located in the gut, such as for us and for mammals and humans, it was the same. And this is why we are all concerned by our gut health, because this is very, very important, but it works for poultry too. And gut health is really crucial to ensure optimal performance for the birds and to ensure a good health and robustness for the birds, to give them the ability to resist to any challenges that they are going to face during their life. So the different roles of the microbiota that have been described are its role in reducing the inflammatory response and also helping the birds to develop its own immunity, whether we are talking about innate immunity with a macrophage pathway or the adaptive immunity with the immunoglobulin production that are going to fight against pathogens. Microbiota is also playing a big role for preserving the gut integrity and the tight junctions between the epithelial cells, and it is also involved in the maturation and the capacity of the bird to develop and for the cells to multiply at the epithelium level. There is also a competition with the pathogens for nutrients, for example, or for oxygens. And this microbiota is playing a role also in the activation of the goblet cells. We have some evidence of that also now, which are responsible of the mucus production, which is the first physical barrier to protect the animal from pathogens colonization like Salmonella. Practically speaking, we know that many factors are affecting the microbiota composition. I may not describe all of them, but you need to know that there are some intrinsic factors related to the animal, like the genetics, the age, the sex of the animal. There are differences between females and males. Usually males are more sensitive, especially during the first days of age. The first days of life are very, very important for the birds, because this is where the birds are going to be in contact with new contaminants. I mean, today in our poultry sector, when the birds are hatching in a hatchery, it's all sanitized, it has been cleaned, disinfected. And the gut is said to be sterile. And this is by having this contact during transfer, during placement on farm, with contact with feed, litter, water, that the bird is going to make its own microbiota. This is where the germs are going to come from. The feed and the water on farm are playing a very big role also in the establishment of the microbiota. So you may look deeply at the raw materials quality, check that you have a correct nutritional balance. Feed presentation, mash is more at risk compared to pellets, because in the pelleting system and the feed mills, you have a heating process that is securing a little bit, doing a little bit of sanitation of the feed. And the water quality, never forget the importance of the water quality. I used to say it's twice more important than the feed, because the birds are drinking twice more than they are eating. Farm conditions are very important, just such as stocking density, ventilation, temperature, pathogens in the farm, like bacteria, viruses, parasites. And don't forget also that the fact that people are manipulating, handling birds might be also stressful to the bird. And it is also having an impact on the microbiota balance. Just an example here, again, don't try to look deeply. The idea is to show you that each raw material is having its own microbial composition. There are some bacteria you will see in some, here in blue, you have soybean meal, for example. Here you have the corn, for example. It is not the same bacteria genera you will find in different raw material. So you understand that according to the feed you are going to give to the birds are going to get a different microbial balance. And just to finish with this part, second part, I want to focus on the effect of antibiotics treatments of microbiota. And this study is really showing what's happening when you use antibiotics in your poultry flocks, especially during the first days of age. The Shannon index is what is showing the diversity of the microbiota. And there they gave, from the starting point to 6 days of age, they gave some antibiotics in the drinking water. And you see that the diversity using antibiotics is really decreasing. So it is all significant. And it takes a long time for the birds to recover a normal level. And in this study, for example, after using amoxicillin, it took 3 weeks for the birds to recover a normal level. So it means you use antibiotics to fight a pathogen, but it has some long-term effect on the balance of the gut microbiota. And the microbiota in natural conditions is really coming from the contact with the parent birds and the environment. So feed, water, hair. And here in this study, a very recent one from 2020, the scientists compared the genera present in birds that weren't in contact with the hens with the group where birds were in contact with some mother hens. And what they have been showing that there were some common genera to the two groups, to both groups, but there were also some specific genera to the two group, test group and control group. And also what they are showing is that the diversity, the number of genera, the abundance is much higher when the birds are in contact with the mother hen. So the profile is more diverse when the birds had some contact with adult birds. And what they've been showing is another way to show the profile of the mother hen. And here you have the birds that weren't in contact with the hen, and on the right, some birds that were in contact with the hen. And simply compare the color, you can see that when the birds were in contact with the hen, they have a similar profile to the mother hen. So it means there is a transmission from the mother. to the chicks, to its progeny, which is not the case when the birds are not in contact with the hens. And there are some missing bacteria that are not transmitted from the mother hen. So let's switch now to competitive exclusion. And what is competitive exclusion? A competitive exclusion product is natural live microflora, which is derived from healthy adult chickens, and it is processed by fermentation. Those type of products are intended to establish, maintain, or restore a good balanced gut flora. And in our modern poultry operations, where I said already that chicks are hatched in controlled environments, the application of competitive exclusion product will play a role of the first inoculum that would be normally given by the parent hens. And this is what is going to give the protection to the birds against pathogens. And coming to the same study, this is completely what they've done here. They gave to those birds that weren't in contact with the hen, they gave them at hatch a competitive exclusion product that have been inoculated to the birds, and you see that the birds are recovering the same profile as the other birds, and which is similar to the mother hen microbiota gut profile. So we can modulate by ourselves and by giving a competitive exclusion product, the microbiota of the chicks, trying to mimic what is happening in nature. What is the definition of the principle of competitive exclusion? It works for all ecosystems, all living species. And the definition is that two species competing for one limiting resource can't coexist at constant population densities. And if we transfer this to our topic about gut health, it is a phenomenon whereby normal healthy intestinal bacteria will colonize the intestine and thus prevent colonization by transient pathogenic bacteria. How it works, 3 modes of action have been described. First, biological exclusion. Competitive exclusion product is going to provide some good bacteria, and they compete for nutrient, but also for oxygen, for example. And so that's, the pathogens won't be able to multiply anymore. The good gut content from a competitive exclusion product is also playing the role of a barrier, and this is what we call the physical exclusion that will prevent the pathogens to attach to the epithelial cells. You have the villi there represented again. So the pathogens are simply not going to be able to approach. And the chemical exclusion is the fact that those good bacteria, beneficial bacteria, are going to produce some metabolite substances that are going to fight against pathogens. I mentioned them already before, short-chain fatty acids or bacteriocins, for example. Here you have an example of the efficacy of such a product, competitive exclusion product, against Clostridium perfringens, which is involved in necrotic enteritis outbreaks. And what they've done in this first study is that they have been doing a Clostridium perfringens challenge at 16 days of age. And prior to the challenge, at day 8 of age, they gave or not a competitive exclusion product to the bird. And what we see that in this group, at day 16, you have the inoculation of Clostridium perfringens, which is a challenge. And the fact that a competitive exclusion product has been given before completely prevented the colonization of Clostridium perfringens in the ileum in this case. And it makes sense with some more recent results from Faker in 2019, measuring the parameters, the incidence of necrotic enteritis outbreaks, whether we are looking at lesion score or mortality. And in this trial, they compared the fact that they were giving a competitive exclusion product once a day old with the fact of giving antibiotics every day in the feed. And we can see there that competitive exclusion product is doing as well, if not even better, than an antibiotic in the feed every day. So it really plays a very efficient role in the reduction of the incidence of necrotic enteritis outbreaks. So you may use a competitive exclusion product to fight pathogens like Clostridium perfringens, but we have some data evidence of efficacy also against Salmonella and avian pathogenic E. coli and multi-drug resistant E. coli. It gives a long-term protection. This is what I've shown just before, just with one application a day old. You can use it in all avian species. And the application is compatible with usual vaccines that are applied at the hatchery, like IB, IBD, you have all the list there. but then you need to take caution with disinfectants or antibiotics that may be detrimental to the live bacteria content of a competitive exclusion product. And these are feedbacks from a company. Oh, sorry, this one moved. A big integration. This is where they started to use a competitive exclusion product, and they are following in this poultry integration the number of birds that are medicated, and you see that there is a huge decrease in the incidence of medication in their poultry integration, and they are still a very good customer of us until now in 2021. And for sure, if you have less bird medicated, you can follow in some poultry integration, you can follow the use of antibiotics. You have different references like milligrams per kilogram of live weight or things like that. And we have exactly the same type of curve, same pattern, with a huge decrease of antibiotic usage when you measure and you refer to this parameter. So some take-home messages related to my talk today is the first, the importance of the startup period. I hope I convinced you that it is really a crucial time for the development of the bird and also for the gut development and the gut health. And a well-balanced microbiota is really needed to support the gut health and the immunity of the bird. And this will help you as a first step to avoid using antibiotics in your poultry operations. And a competitive exclusion product that will be given as soon as hatch will provide to the chick the optimal gut microbial content, and this will give them the ability to resist to pathogens colonization throughout their life, whether we are talking about Salmonella, E. coli, or Clostridium perfringens. So now, if you have any questions about my talk, I would be pleased to answer them. Thank you.
Okay, thank you very much, Dr. Morvan. Very interesting presentation. I think by now people are just trying to connect brooding systems, temperature with regards to gut development, and how it's very important in broiler production. So thank you very much. At this moment, we'll start with the third paper, and this will be presented by a veterinarian, Spanish veterinarian, and his topic is all about Global Hatchery Health Program, Practical Experiences on Supporting Day-Old Chick Health. Our speaker this afternoon is a product manager with the Poultry Business Unit at IPRA. He graduated from the Autonomous University of Barcelona in Spain with a degree in veterinary medicine. He had his internship on the grandparent stock management at Envisions, Spain in 2018. He has taken positions in technical services and sales associated with biological products and feed additives. He recently joined IPRA, specializing in Salmonella killed vaccines and technical services focusing on improving day-old quality at the hatchery level. So mga kaibigan, let's welcome Dr. Santiago Yago de Castro Verges.
Hello, good day everyone from Spain. My name is Santiago de Castro. I'm a doctor of veterinary medicine, and I'm currently working at IPRA as a corporate product manager, being responsible of our killed Salmonella vaccine, Navisan Secure. I bet that most of you may know of this product, and also for one of the latest technical services we have launched recently this year, as it is the Global Hatchery Health Program. Before start with the presentation, I would like to thank to the PCPP for organizing such a technical event, and also thank for the consideration and the chance they gave me to briefly expose these practical experiences that we have helped our customers in improving the day-old chick health and quality, and also to improve the profitability of their chicks through these evaluations and correction of the vaccine procedures, day-old chick quality, and finally, the study of the database collected at the hatchery level. Because most of you, if you are attending at this presentation, really worry about your chicks, worry about the performance of the hatchery, and always want to improve and be the best on your field or your business. And it's for that from IPRA, we created this service to maximize the support that we can offer our customers in these goals. So what is it? Why is the Global Hatchery Health Program made for? How a global hatchery health program is performed? Let's see it. This service comes from the need of our customers of having the total control of the hatchery. Okay, so for this purpose and our expertisement in the vaccine field, we decided to implement a service that first guarantees the highest quality of the vaccine, because if we have a quality vaccine, but the application is not good enough, we are decreasing the efficacy of the vaccine, or said with other words, we are throwing away our money. Then we thought, okay, we are guaranteeing our day-old chicks a high quality vaccine with a high quality application. Nevertheless, sometimes if there is a slight failure during the incubation process or inside the breeder farms as well, the quality of the chick can be impacted and decrease slightly. So we decide to go further and also check the quality of these birds and provide a support to our hatchery managers in this matter. So inside this block, inside this stage, we also analyze the birds physically, but also microbiologically. Okay, as you may know, there are some bacteria that can be transmitted vertically and can impact heavily the performance of these young birds. So we also decided to provide an extra support in this matter by analyzing the microbiological quality. Okay. Finally, some of you also have tested our great database analysis service called IPRA-Stats, worldwide recognized for its help and support to customers in detect areas of improvement. And now we have adapted also this service to the database collected inside the hatchery. Okay, so with all these three services based on the quality of the vaccination procedures, day-old chick quality, and the key point analysis, day by day are tens and lots of hatcheries that are actually improving its performance through the Global Hatchery Health Program. Now, going deeper on every block, we will make a review on the points to analyze one by one. Okay, so first point is to check that our vaccines are in the proper storage and conservation conditions. Of course, temperatures must not underpass 2 Celsius degrees, and must not overpass 8 Celsius degrees. Any variation out from this range will affect the quality, but also the efficacy of the vaccine. In my opinion, it is also recommendable to have a maximum and minimum thermometer, okay, and make a daily register. Okay, in that order, if someday, for example, suddenly the lights go off, you can notice by a fast increase in the fridge temperature. Okay, so take in consideration these recommendations. Okay, take into account also, please, that I always say that vaccines must not never touch directly to the wall of the fridge. Okay, because the walls are much cooler than the inside, and we have the risk of freezing. So, okay, and last but not least as well, we want to give the vaccine into the best conditions. Okay, it's for that we must not put any kind of food or drinks into the fridge that may contaminate the vaccines. Okay. Moving one step forward, we see the materials and the preparation. Okay, so the space where we prepare the vaccine must be a clean and disinfected room, of course, with also good illumination and nice ventilation. Okay. In addition, of course, all the materials that came into contact with the vaccine must be disinfected as well. It is also recommendable that this kind of procedures should be done always by the same worker, okay? Because if we recommend to do it to several workers, maybe one worker does it on his own way, another worker does it on his own way, and there are maybe failures that we can never assess. Okay, so it is recommendable that just do this kind of procedure the same person always. Okay, so vaccines that are mixed with water must pay special attention to the quality of water, of course. Okay, so please make sure that the water that we are using is chlorine free, okay, and also free from any sort of disinfectants. Okay, I would recommend to use bottled water, okay, because you are aware of what you're using, and sometimes when you use tap water, it can be also disinfected with some kind of chlorine. So pay attention to this feature. Once all the materials are ready, it is paramount also to calculate, okay, the properly volume of water that we are using, okay, for a specific amount of birds, okay, that we want to vaccinate. For this reason, especially in sprayable vaccines, you must check also the volume of the vaccine that's priced for each box. Okay, it's not the same if we dilute the vaccine with a spray machine that sprays 24 milliliters for each box than another machine that sprays only just 14 milliliters. So always verify that the water volume fits with the number of birds you want to vaccinate. All right. As I've said, next step is to check the status of the device, the machine, okay? Whether it is a subcutaneous or maybe it's a innovo machine or a sprayable device, all of them must be checked daily, okay? You know better than anyone that every day thousands of chicks are vaccinated. So pay attention to the devices, to its status, to its dose accuracy, to the cleaning, also in innovo and subcutaneous, the quality and the length of the needle, okay? And for sprayable vaccines such as coccidia, bronchitis, check the proper distribution among all the box, okay? Check the droplet size as well. And most of the vaccines applied require a droplet size categorized as coarse spray, okay? Then finally, it comes to the application stage. At this point, we separate by the route of administration. Starting from the innovo evaluation, is very important to vaccinate into the amniotic liquid, okay? And for Cumboros vaccines, embryo is also permitted, okay? Embryonic stage recommended for this vaccination is among 18.5 days and 19.2 days, okay? Failures at this stage usually come from the needle length, or maybe because the machine is not adaptable to the size of all the eggs, okay? And if innovo vaccine is not permitted, subcutaneous is another way to vaccinate our birds at a day old. Okay. At this stage, they all is always important to assess our operators, because in function of the position they set the bird in the machine, the vaccine will be delivered properly or not. Okay, so please every day assess your operators regarding this issue. Okay, for this reason, it's also recommendable to add authorized colorant in order to check the vaccine location. Okay. Once we leave behind the vaccine block, we focus on the day-old chick quality control. As we commented before, best vaccines for express all of its potential must be applied to the best chicks. One of the most practical and most known worldwide methods is to assess the chick quality by Pascal score. Okay? This method quantifies the physical defects that can clearly be seen, like navel defects, leg irritation, maybe also dehydration, also peak irritation, abdomen abnormalities, and finally vitality feature as well. All these mistakes are quantified, and we finally get a final score that provides to the hatchery manager useful information tools about the incubation process. For example, a high rate of problems on the vitality may come from higher temperatures at the hatcher or even insufficient ventilation. Okay. If we want to provide high quality chicks, we must take into account also the microbiology quality. Okay, inside the hatchery, we find high density, we find high temperatures, and also we find high humidity. Okay, that provide a perfect environment for the multiplication of different bacteria, and especially those ones that can be transmitted vertically, such as Mycoplasma asepticum, Mycoplasma synoviae, E. coli, also Salmonella. Okay, so inside our technical service, we have given special attention to this evaluation. We have created FTA cards for the detection by PCR of these 4 bacterial species. By the analysis of the day-old chicks, we can anticipate possible field challenges that may occur during the fathering, but also determine which breeder flock is contaminated and try to tackle this contamination. Keeping in this context, the context of prevention, as you know, in IPRA, we give a paramount and a crucial importance to prevention. For this reason, we consider that being able to understand all the data collected will also help in prevent possible mistakes or failures during the incubation process. So inside the Global Hatchery Health Program, our database analysis service, IPRA Stats, performs a deep study of the failures that can affect and alter also the hatchability and first week mortality, okay? Because it is difficult to manage or handle these infinite and large tables of data. So in IPRA, we have the support of prestigious database scientists that during the last 2 years have helped poultry companies all over the world to detect areas of improvement that were unknown before. Okay, so now that all the theoretical part has been explained, I wanted to show you a few of the tens of cases that we have seen during the Global Hatchery Health Program evaluations, and thanks to that, improve the quality of the chicks and also the profitability of our customers all over the world. We will see 2 practical cases of each block of the GHHP. Okay, GHHP is the abbreviation for Global Hatchery Health Program, so is the short name for this long program name. Okay, so starting with the first case, I'm going to show you is from a customer from Russia. Okay, these were a broiler integrator of approximately 2 million broilers per week. Okay, so among different IPRA vaccines they were using at that moment was Gumbo Hatch, okay, our Gumboro vaccine for de novo or subcutaneous. In this hatchery, especially, they decided to apply the vaccine in de novo route. Okay, so once they started vaccinated with Gumbo Hatch, one of the farms where the field surveillance was done, they were kind of surprised because no IVD vaccine strain was found in all the birds sampled. Okay, so it was kind of strange because if the vaccine is applied de novo, you would expect a greater amount of positives to the vaccine strain. So it was decided to check the vaccine application at the hatchery. For sure, colorant was added to the vaccine preparation to verify the site of injection. Okay, that is a paramount evaluation in this kind of audits. So here are some of the pictures, images that were taken during the de novo evaluation. Okay, 60% of the birds were negative to the vaccine just the moment after the application. Okay, so that induces a failure in the application of the vaccine. Okay, the needles were checked first and all of them injected vaccine. So there was a problem with the device. Okay. After analyzing deeply the Innovo device, it was notified that the Innovo device could not apply correctly the vaccine to those especially smaller eggs coming from younger breeder flocks. So in this situation, the recommendation was to adapt, was to check the machine by the operators, okay, update it if they could for improving the quality of protection of those breeds coming from smaller eggs, and if not possible, separate the eggs from young breeders and try to vaccinate them via subcutaneous. Okay, and last suggestion, which the customers are considering currently, is to use a device, a Innovo device, with a more practical adaptation to all the egg sizes. Okay, that is, in our opinion, the best way to improve the quality of the vaccination. Okay, so second case, we also focus on the application of the vaccine. Okay, is this one from a broiler breeder customer from Korea, from South Korea, and one of the latest breeder flocks acquired had a sudden increase of deaths that respond good to amprolium. Okay, so on the necropsy findings performed by the IPRA technical services, there were a clearly cecal necrosis that was found. Okay, samples were also sent to the diagnosis lab, and they found a Meriatenella, okay, as the cause of the lesions and the deaths. Okay, we decided to check the application. And what did the technical service found? Let's see if you can guess the failure on the application. Okay, this is a picture taken just 10 minutes after the spray of the vaccine. Okay. Well, as you may have noticed, not even a 40% of the birds were vaccinated properly. Why? There was a recently change of the course spray device on the breeder's hatchery, and wasn't checked yet for the right functioning. So the device didn't cover all the box, and most of the birds didn't have time to ingest the vaccine. So please check always, pay special attention to the application of the vaccines, okay? Because we can throw away a lot of money if we don't do these evaluations. So the third case, case number 3, this is regarding to the day-old chill quality. In this case, as I mentioned, we agreed with the customer to perform the GHHP once a month. And in one of the PASGAR score evaluations, on the physical evaluation, we noted something strange. Okay, on the first flock that we evaluate, was day-old chicks coming from breeders from an age of 54 weeks old, okay, a bit older. A large number of chicks, we found that they had open navels, okay, and unlike the other breeder flocks, which navel ratio effect was under 6%, this first flock had 40% open navels. So something was really wrong done here. Thanks to the Excel data sheet, we can clearly assess that the difference between the flocks and also the defects of each flock. Okay, in this case, the solution was as simple as switch the order of the hatching chicks. So this breeder flock had few more hours to close the navel. Okay, you know that the older the breeder gets, the larger the poults are. So few more minutes of incubation are sometimes recommended for those eggs coming from older breeder flocks. Moving to the fourth case, here we had to deal with a bacteriology issue, okay, of a customer who was suspecting from different bacteria in 2 breeder flocks, whose broiler had higher mortality rates than IPRA diagnosis, provide them this microbacterial analysis through the GHHP. The day OC quality by Pasgar score was also assessed, just to discard any incubation issue. And after taking samples from all 6 breeder flocks for Mycoplasma rheizecticum, Mycoplasma synoviae, E. coli, and Salmonella, finally, just E. coli was found in 1 breeder flock. Okay, from this positive flock, antibiogram was also done. That case, that the birds started with early mortality, and otherwise, parent stock positive from E. coli was also prevented with a higher biosecurity and nutrition strategy, including organic acids during 2 weeks, disinfection at the arrival at the hatchery as well. And after this service was done, the mortality rates from the bulls coming from this parent stock decreased until at 2.8% in the next flocks. So in my opinion, it's very important to keep monitoring of microbiological quality. And in IPRA, we are glad to give this service to our customers that definitely support on their daily basis. Finally, as the latest practical experience, we had one customer that wanted to make a trial with one of our vaccines, and also a database analysis was offered. So this hatchery had thoughts that the multi-stage hatchers didn't seem to work as efficient as the single-stage hatchers did. So IPRA Stats collected all the data from the last year and performed a deep analysis, taking into account all the features that could impact negatively to the hatchability of the eggs and to the first week mortality. Okay. Surprisingly, one of the first results that we extracted was that, well, in fact, the hatchery manager didn't notice, were that after studying the hatchability from all the breeder flocks and after classifying them by the breeder age, we discovered that 2 of their breeder farms had very bad results in all of their ages. Okay, so the breeder technical team could also focus on improving, especially on those farms. Okay, then focusing on what they were worrying about, there was a slightly higher first week mortality on those eggs that were coming from the multiple stage. Okay, that was the hatcher where they were placing the smaller eggs of all the hatchers. So, in summary, after this presentation, what I would like you to keep clear is that inside the hatchery is complicated, is a little bit difficult to have everything under control. So external services as the GHHP give a crucial support in ensuring the quality of the vaccine application. So we are enhancing, we are boosting the protection of our birds. Okay, it is important always to anticipate field challenges, be able to know where we are, where are we going to, what do we want to achieve. Okay, so for this reason, evaluations such as Pasgar score and microbiological evaluations are so important for taking the control of all the biological and mechanical procedures that take place inside the hatchery. Last, please be aware of the importance of database management, as a few cents of US dollars, as you know, can make an operation profitable for the company or a complete disaster. So try to get the excellence, try to get the maximum always, and for sure, we will be delighted to help all of you in getting this goal. Thanks all of you for your attention, and special greetings to Esmeraldo and Mildred for your consideration, and thanks for your commitment in raising awareness regarding the importance of the control of vaccination, DOC quality, and database management. So thank you, all of you.
Maraming salamat, Dr. Yago, for your very good presentation about making sure that we have excellent sanitation at the hatchery, and very important because this affects how our broilers are going to be raised at the farm level. Okay, so thank you very much. Now I'd like to introduce the fourth speaker, who will be presenting on the strategic applications of beta glucans from sugarcane molasses yeast in broiler diets for sustainability. Our speaker this afternoon specializes in animal nutrition. She is a graduate of Bachelor of Science in Agriculture at the University of the Philippines, Los Baños. She also earned her master's in animal science, majoring in animal nutrition, also at the University of the Philippines, Los Baños. She took her master's in business administration from Ateneo de Manila University, and now is currently on her doctorate program for business administration at the De La Salle University. She has gained a broad business acumen from managing her own company for almost 2 decades. She is an active member of the Philippine Society of Animal Nutritionists, or PhilSAN, and is a certified digital marketer of the National International Marketing Mobile Association of the Philippines. Ladies and gentlemen, let us welcome the Chief Operating Officer of JA Farms Asia Pacific, Ms. Arlene M. Opelano.
Good afternoon. Welcome to our 2021 Poultry School Online. I'm Arlene M. Opelano from JA Farms Asia Pacific Inc., and I'm here to share with you the strategic applications of beta-glucans from sugarcane molasses yeast in broiler diets for sustainability. Our topic right now is practically very timely with our current scenario. Resilience. Resilience is the ability to adapt appropriately towards the effects of a problem. And the emergence of uncertainty causes organizational resilience to be tested. They face specific challenges and difficulties of spatial and civil remoteness. OR, or organizational resilience, as a phenomenon that can occur with varying degrees of intensity affecting their resilience per se. It can be stabilize, change, or exacerbate the state of an entity subject to adverse external force. Resilient people and organizations possess 3 characteristics: a staunch acceptance of reality, a deep belief, often buttressed of firmly held values that life which is very meaningful, and uncanny ability to improvise and undertake activities. Resilience is indeed coming from within. This is composed of 3 factors. We have resilience from an individual, resilience from the organization, and behold, resilience for the community. Now let's take a look on the strategic nutritional model that we have for our poultry operations, specifically broiler nutritional diets. As you can see, we still have to give core focus on the food safety in order for us to have a very good BPI index under Philippine condition. And beta-glucans indeed gave us a very consistent results in terms of production performance, whether it's coming out from a very good BPI of around 390 to 400 nowadays is indeed very timely on how we can operate efficiently. Not to mention, of course, mycotoxin absorption, pathogen binding, and immunomodulation brought about by beta-glucan. But when combined together, we have specific sets of feed additives that we can take a look to. We have bactericidal, we have bacteriostatic, we have gut microbiota that we need to consider, anti-inflammatory, and antioxidant, and specifically the mycotoxin absorption within our diet. Now, let's make a review. What about the effect of dietary beta-glucan supplementations under heat stress? So we're talking about growth performance, nutrient digestibility, and ileum microbiota. For growth performance, there were several papers and studies that it increased the body weight gain, the feed intake, and the FCR. For nutrient digestibility, This is being manifested by the body weight gain increase, the feed conversion ratio, statistically at P, which is greater or less than 0.05, and greater energy digestibility. But when we talk about ileum microbiota, this would increase number of goblet cell growth and villus maturation, cure of villus damage or loss caused by the pathogen challenges inside the ileum, and of course, there is an expectation of a decrease in E. coli amount in the ileum and an increase in gut health for higher digestibility and of course, increased in feed conversion ratio. There is also a study made by Zhang et al. Of 2020, which is very recent, that we had an increased level of TNF, interferon gamma, interleukin 1, by heat-stressed exposure of these birds. Definitely, this will decrease level of TNF-A in heat-stress condition within the farm. Now, with this level, we also identified increased pro-inflammation cytokines and increased with a very high ambient temperature. For this, with regards to immune profile, we also discovered, studies show that there's an increase in passive heat exposure and increased leakage of endotoxin from the intestine to the systemic circulation of the blood. More so, there is an increased activation of leukocytes and endothelial cells and an increase characterized by the release of these pro-inflammatory cells and anti-inflammatory cytokines. This was cited by Brookhammer et al. Of 2002. Now let's take a look on the antibiotic side effects in animal health. This showed that with the use of the beta-glucan per se, we can make sure that the thickest cell wall coming from a more mature cells would give us a relatively high performance in terms of production index or broiler production index. Why it happened? You can see the tight junctions with this, but with the use of these beta-glucans, definitely it activates the cytokines. It will not allow the pathogens to multiply for so. Thus, it will stop the proliferation of the different kinds of the pathogens. Beta-glucan is a natural solution wherein you can enhance animal productivity and maximize profitability of the farmers. For years, we discovered the consistent results of beta-glucans. We already saw it with the different species at hand, all peer-reviewed journals. But let's take a look on the more applicable side of this data. Reduction in pathogen agglutination. We were able to quantify that 99.6 and 99.4% it can agglutinate Salmonella and E. coli respectively. How much more? We can even eradicate the usage of anti-Salmonella in our diet formulations. Okay? On the other hand, for intestinal integrity, we were able to see different sets of the materials that we used when subjected to different kinds of scenario for the birds per se. We maximize this for maintenance at least 2 kilos per ton, or at heat stress related activities, we can still start it at 0.5 to 1 kilogram per ton. Now, with regards to immune system modulation, this is very important, wherein we were able to see, observe results in terms of supplementation because there is an increase of goblet cell growth and villus maturation and cure of villus damages caused by the pathogen challenges. There is a decrease in E. coli amount in the ileum and an indirect evidence of better gut health for higher digestibility and another factor for FCR improvement for these stressed animals or stressed birds. One of the major consistent results that beta-glucans had given us through this year is the reduction of mortality. Reduction in mortality, whether it's layers or broilers. This would complement on its mode of action wherein it activates the cytokines. Now, with regards to consistent agglutination capacity, as you can see, since 2010 to 2016, we were able to quantify how many or what about the meta-analysis of these data results. We can see the 99.6% agglutination for Salmonella and 99.4% agglutination for E. coli. Of course, we all knew the mode of action as it activates the cytokines and release a specific action for the macrophages to take its effect. Now, in nutrition, it's very important for us to quantify that beta-glucans coming from sugarcane molasses yeast will not be digested inside the animal. As you can see with the numbers, the beta-glucan 1 and the beta-glucan 2, it has a very low digestibility percentage, which is around 53.1 and 65.4 respectively. As compared to the Baker's yeast cell wall 1, 2, and 3, it just gave us 85.8, 74.6, and 73.7. What does this mean? This meant that the Baker's yeast cell wall coming from these beta-glucans were used as toxin binders per se. But with beta-glucans coming from sugarcane molasses yeast, which is coming from beta-glucan 1,3 and 1,6 linkages, it has a different function in the formulations or in the performance of the animals. These were all used as performance enhancers. So remember, we're talking about digestibility, and digestibility of these beta-glucans should be very low since it is coming from the thickest cell wall of around 100 to 200 nanometers thick. Now, let's take a look on this, a very good review on the light microscopy of two kinds of the beta-glucans. Beta-glucans coming from sugarcane molasses yeast, and the other one is coming from primary yeast or brewer's yeast or baker's yeast. As you can see, the beta-glucans coming from sugarcane molasses yeast has a very thick cell wall. Okay? Now let's take a look on a more practical side on how to maximize these beta-glucans for broiler applications in our animal nutrition. We conducted a study with regards to the effect of dietary supplementation of beta-glucan and nucleotides either alone or in combination with AGPs in the broiler diet. This is composed of 6 treatments. As you can see, we have the negative control plus the commercial broiler diet without antibiotic. The treatment 2 is a nucleotide with AGP, okay, with at least 500 mg per kilogram for the AGP. On the treatment 3, we made use of the nucleotides and combination with regards to the AGP used. Treatment 4 is composed of nucleotides with the beta-glucan and AGP. And of course, treatment 5 is just pure nucleotides. And treatment 6, we have combination of the two. What's the effect of the nucleotides and the beta-glucan? Okay? Remember, beta-glucan will block the proliferation of your pathogens. But in terms of renewal of these hepatic cells, you need adenosine triphosphate, which is coming from nucleotides. Let's take a look on the result. We discovered that the body weight, feed intake, and feed conversion ratio of broilers at different periods of the experiment showed that the combination of the nucleotides and the beta-glucans and the AGP performed better among any other treatments. This is around 1.43 for 1 to 28 days, and upon harvest, this is around 1.742. Not to mention the body weight and the feed intake. On the other hand, we also tested vaccine titers against Newcastle disease and infectious bursal disease or IBD at different periods of the measurements or the experiment. And it showed that IBD monitoring performed better with regards to the combination of our nucleotides and beta-glucan and AGP. Of course, there is also a very good number coming in from nucleotides per se of around 3.611. And nucleotides plus beta-glucans gave us a very good result at 3.693 for IBD vaccine titers. Remember, we're talking about p-value here of around 0.008. Let's take a look on another set of the experiment. This is intestinal microbial counts for 40 days age of monitoring. What does the table give us? This gives us a clear picture that the usage of our beta-glucans would give us a very good result in terms of E. coli agglutination capacity, which gave us at least for 8.21%. Okay? Now, let's take a look on the fourth side of the experiment. What does this mean? Generally, it has been observed that beta-glucan and nucleotide and AGP, when supplemented together with the villi height, increased in the duodenum and jejunum at P 0.05 value. What does this mean? This means that with duodenum and jejunum, we generated very good results with these sets of combinations. Now, another study was conducted with beta-glucan immunomodulatory and intestinal integrity effects on broilers challenged with Salmonella enteritidis. We all knew here in the Philippines, it has been a standard for us to monitor this Salmonella program inside our diet. But this time, let's take a look what can be the standard out of the beta-glucan usage. This is composed of 4 treatments, control plus the Salmonella treatment. The second treatment is with beta-glucan alone, and the third is control alone, and the fourth would be beta-glucan with Salmonella. Let's take a look what happened with the experiment. This classical picture gave us an idea that the intestinal integrity is ensured by a continuous layer of epithelial cells with our tight junctions, united by tight junctions per se and by mucosal layer above the epithelium. Permeability of intestinal mucosa played a big role in terms of monitoring with this one. And we can see how it goes with regards to the evaluation of the intestinal integrity of the birds. The higher values indicate higher permeability, which is indicative of lower mucosal integrity. Different letters on each group indicate statistical relevance. Differences were calculated by one-way analysis, or what we call the ANOVA, but of course with the use of our Tukey test, multiple comparison test. At 4th day old, the challenge group presented the highest passing marker in the blood, indicating lower intestinal integrity. This effect was mitigated by the use of the beta-glucan, and the control infected group remained the group with the latest integrity at day 8, although there was only statistical significance against the group 2 at this age. In the other collection points, intestinal integrity did not vary between groups among treatments. Now let's take a look on IgA anti-salmonella of broilers challenged with Salmonella enteritidis. What does this mean? Disinfected animals present more APCs than the uninfected and untreated control due to the presence of Salmonella or the yeast cell. Activation of monocytes is the number of cells that are active, but Salmonella invades monocytes by reducing their efficiency. Now, in terms of IgA, we can see that the evaluation of the Salmonella gave us the plot with relative quantification of IgA in the reactive serum of LPS of the bacterium. The cutoff line is represented, but with different letters on each group indicates statistical relevance. Now, the amount of specific IgA against Salmonella was evaluated in blood sample at day 14, but the infected and treated with beta-glucan presented the highest number of Salmonella with IgA. Now, what happened? The infected group, in comparison, had 3 of 7 reactive animals, showing that the specific response of the immune system was faster and stronger, consuming less energy and nutrients since the inflammatory response appeared to be shorter. Now, there's a statistical relevance indicated by these letters on each group. ANOVA test was indeed applied, but the suppressor monocytes prevent an unrestrained immune response, and the beta-glucans plus the Salmonella provided more expressive anti-inflammatory response at day 14. Okay, so this result may be related to the reduction of the inflammatory state of the gut and balance between pro and inflammatory response. Activation of these monocytes in the amount of the cells are very active, but with Salmonella, it invades monocytes by reducing their efficiency, and this can be observed when comparing the infected groups among each treatment. This is very new to us, especially for nutrition, and it's very important for us to understand how this works together with regards to the response with Salmonella-treated treatment. The helper T lymphocytes is the sum of peripheral lymphocytes and mucosal lymphocytes. The mucosal T lymphocytes interacts with beta lymphocytes to produce IgA, and statistically, they have a relevance in response by different letters on each group. The suppressor monocytes prevent an unrestrained immune system. So at day 14, it provided a more expressive anti-inflammatory response for this matter. You can see the difference in the performance of the beta-glucan with regards to mucosal cytotic T lymphocytes and an activated cytotic T lymphocytes as well. Now, an IgA anti-salmonella is indeed very important for us to monitor. What does this mean? This means that the evaluation of the anti-salmonella IgA, the plot shows the relative quantification of IgA in the reactive serum against LPS of the bacterium. The cutoff line is represented, but the statistical relevance is indicated by different letters of each group. Now, look at the graph. The amount of specific IgA against Salmonella was evaluated in an excreta sample at 14 days of age. The infected and treated with beta-glucan group presented the higher number of reactive animals, as well as the highest average amount of anti-Salmonella IgA. The infected group, in comparison, had 3 of 7 reactive animals, showing that the specific response of the immune system was faster and stronger, consuming less energy and nutrients, since the inflammatory response appeared to be shorter. Now let's take a look on the ileum villi histopath at 14 days of age. So we were able to monitor the average area, okay? So evaluation of the anti-Salmonella IgA, the plot shows, definitely gave us a very good picture on this one. The evaluation of anti-Salmonella IgA, the plot shows that the relative quantification of IgA in the reactive serum against LPS of the bacterium. The cutoff line gave us a very good picture wherein we can see the usage of the beta-glucan. Now, with regards to the goblet cells, we had 4.62. With regards to heterophils, we have 0.75. With ILP, we have 9.6. And 4.28 with the average area of the LP for Zika villi histopath. When we're talking about lymphocyte count of immunofluorescence, with regards to liver histopath, we were able to discover the importance of this area. Now, for liver histopathology, there was no statistical difference between the contaminated groups. However, the beta-glucan supplemented group increased the amount of circulating immune cells in the liver, the same way as the results for your immune cells in the blood. Now, for lymphocyte count by immunofluorescence, you can see the liver results confirm the histopath where the challenge G1 and G4 group increased the amount of immune cells. If we correlate with the data for goblet cells, we observed the G4 group presented the highest production, presuming a better lesion prevention and inflammatory infiltrates, which reflected the lower lymphocyte count in the Zika. Beta-glucan is indeed decreased the number of phagocytic cells with the highest number of reactive cells and highest level of anti-Salmonella and IgA. The challenge induced marked inflammatory responses in the intestine and liver and assessed by our lymphocyte counts, section area, and goblet cell counts together with our tissue architecture. Now, in terms of economic implications, what we can see is an ROI of 5:1. To sum it up, the results of our data, the effect of dietary supplementation of beta-glucan and nucleotides either alone or in combination with an antibiotic growth promoter in broiler diet increased the villi height, duodenum, and jejunum. And the beta-glucan immunomodulatory and intestinal integrity effects of broilers challenge with Salmonella enteritis was very effective in controlling leukopenia and prevented immune subset fluctuations provoked by APCs and cytotoxic cells. So nowadays, under our most challenging times in our operations, we really have to be resilient. We really have to be open-minded and robust in terms of execution. What will be the next program for all our operations? And this strategic nutritional tool with the usage of beta-glucan will really help our operations to hit the bottom line. The BPI index of around 380 to 400 is indeed very actual, with the basic foundation on the first 7 days of hitting the birds at 180 to 200 grams per day. So for that, thank you very much for joining us in our journey as we continuously provide safe feed for safe food for all Filipinos.
Maraming salamat, Arlene. Very good presentation, as always. Very detailed, a lot of studies. Hopefully, everybody will have some more information about the beta glucans. Okay, so let's move on to our last speaker this afternoon. And this speaker will talk about dietary selenium forms and drivers of efficacy, from genes to biological effects. Our fifth speaker this afternoon earned his doctorate in animal nutrition at the University of Turin. He was a speaker at the recent Animal Feed Manufacturers Association in South Africa in 2020. And he has been a regular speaker at the Australian Poultry Science Symposia in 2019, 2020, and 2021. He has been an author and a co-author of more than 30 peer-reviewed technical papers. Currently, he's working as the Global Technical Manager at Adiseo France SAS. Mga kaibigan, let's welcome Dr. Michel DeMarco.
Good morning and good afternoon, everybody. So my name is Michele De Marco, and I'm a Global Category Manager in Adiseo. So today I'm very glad to be here and to have the chance to talk about one of my favorite topics, which is selenium, and in particular about dietary selenium forms and driver of efficacy, from genes to biological effects. So let's get started. So as all of you know, living organism relies on oxygen because oxygen is the fuel of life. So normally oxygen is used by mitochondria in the cells to produce ATP, and the normal outcome is water. However, in a specific condition, those mitochondria can become less efficient. Therefore, there is a leakage of electrons and the production or the synthesis of the so-called reactive oxygen species, which are, for example, superoxide, hydrogen peroxide, and hydroxyl. And it is also well known that those reactive oxygen species can be, in some cases, good, but also very bad if they are not well controlled. In fact, in living organism, in a physiological condition, we are always in a reversible zone, where we have from one side the production of these free radicals, and on the other side the antioxidant systems which are able to maintain the redox homeostasis. So as we said, those free radicals can be in some cases good and having positive effects as signaling molecules. In fact, in these slides you can see two examples. In the first example here in the left, you can see a protein composed by two cysteine that, depending on the surrounding redox environment, can be reduced or oxidized. And this on-off switch lead to structural changes of the protein, making this protein mobile or not, activated or not, making, for example, this protein an active enzyme or not. And this is a clear example of an activation or inactivation of an enzymes. And in this other example, in the right side of this slide, we have two proteins that are combined together, that again, depending on the redox environment, those two proteins can be combined together or separated. And this is the classical example of transcription factors, such as, for example, the NF-kappa B or NFR2 transcription factor, which are involved, for example, in the synthesis of dedicated genes, for example, in the immune and inflammatory response, where we have the synthesis, for example, of pro-inflammatory or anti-inflammatory cytokine. So we can see that here in this case, free radicals and the reactive oxygen species are really playing a signaling role to modulate different metabolic and physiological responses. Then those reactive oxygen species can be good and bad at the same time. And an example is, for example, what is happening in the phagosome, in the phagocyte cell. When we have a microbe, the phagosome is capturing the microbe and is starting to produce high level of free radicals, which are used as a weapon to kill the microbe. However, when this production became very important, those free radicals can escape the phagosome, starting to have a detrimental effects on the healthy tissues around. And then last but not the least, those free radicals can have very detrimental effects in a different level. In fact, when the synthesis of free radicals became very important and the antioxidant system are not enough to maintain this redox balance, those pro-oxidant molecules lead, for example, to lipid oxidation, in particular in the cell's membrane, which are rich in polyunsaturated fatty acids, can oxidize protein, making those protein inactivated or creating protein aggregation, and can also impair and having a detrimental effects on ADN, causing ADN oxidation, causing, for example, genes codification error. So to handle with these reactive molecules during their evolution, animals, they have developed and shaped the antioxidant systems. These antioxidant systems are like a football team with many players having as a main goal to maintain the redox homeostasis. So in this antioxidant system, as I say, we have many players. We have, for example, vitamin E, defined as the headquarter of the antioxidant system. We have carotenoids, flavonoids, vitamin C, the gene expression and the synthesis of different antioxidant enzymes such as superoxide dismutase and catalase. And then we have selenium that has been defined as the chief executive of the antioxidant system. And now I will try to explain why selenium is so important. So when we talk about selenium, first we need to have a look of the periodic table. In fact, as we can see in the slide, selenium belongs to the same group of sulfur and oxygen. Why? Because similar to sulfur and oxygen, selenium has this specific capability to exchange electrons. So another thing that we need to remember is that selenium is biologically present in living organisms as two selenium amino acids, selenomethionine and selenocysteine. And overall, to summarize, we can say that there are 25 genes which are coding for 25 selenoproteins. So as I said, selenium is present in the body as a two selenium amino acid. We have selenomethionine, which is a safe storage form of selenium. As we can see in this slide, we have methionine and selenomethionine. And as you can clearly see, the only difference between methionine and selenomethionine is that in the selenomethionine, we have a selenium replacing a sulfur. And these specific characteristics make selenomethionine a molecule which can be unspecifically incorporated in body proteins by replacing standard methionine, because the body cannot make the difference between methionine and selenomethionine. Therefore, when animals are fed with selenium in the form of selenomethionine, are able to create this body storage of selenium, something that I like to call a selenium insurance policy. And then we have selenocysteine, which is the true biological active form of selenium. So selenocysteine has two specific characteristics. The first one is that it has to be de novo synthesized by the body. And the second one is that being the active form of selenium, selenocysteine is present at the catalytic site of each selenoprotein. In fact, in each selenoprotein, there is at least one selenocysteine, which is providing the enzymatic capability, so the capacity to exchange electrons to catalyze different reactions. So the selenoprotein synthesis rely on the selenium supplementation through the diet. In fact, when we provide animal different dietary selenium compounds, those selenium compounds are then transelinated in the liver, so are transformed into hydrogen selenide, which is the precursor of the synthesis of de novo selenocysteine, which is then bounded at the catalytic site of the selenoprotein, which are mainly antioxidant enzymes. And when we talk about selenoprotein, we indeed talk about genetically encoded protein. Today have been discovered 25 selenoprotein in mammals and 26 in avian species. As I said before, each selenoprotein contains at least one selenocysteine at the catalytic site of the enzymes. They are very sensitive on the overall selenium intake, and they have many different functions in the body, while the majority of them playing a very important role in the redox balance in the body. And in this slide, I just reported some function of a selenoprotein. As you can see, the majority of them are antioxidant, playing an important role in the redox balance and as signaling molecules, but they are also involved in other functions, such as the thyroid hormone metabolism, the protein folding, lipid metabolism, selenocysteine synthesis, selenium transportation within different parts of the body, calcium regulation, and also involved in the spermatozoa maturation and function. And for example, we have, as I said, different selenoprotein. And here I just reported a few examples. We have, for example, the glutathione peroxidase family that they have a specificity of activity. In fact, we have different types of glutathione peroxidases. We have a glutathione peroxidase 2, which is highly expressed in the gut. We have a GPX4, which is highly expressed in the testes, providing male fertility. GPX3 that is highly expressed in the plasma, and then we have GPX1 that is expressed at many different levels in the body. And glutathione peroxidases are very important because are involved in the degradation of hydrogen peroxide, which is a free radicals, into water, which is a molecule that is no more reactive. Another example are the deiodinases, which are modulators of the thyroid hormone synthesis. In fact, these three selenoprotein are involved in the activation and inactivation of the thyroid hormone T3 and T4. And these thyroid hormone are implicated again in many different functions, for example, in the lipid metabolism, in the thermoregulation, and in the control of the growth and the development of the animals. So we understood how selenium is important. So now the question is, which source we need to supply to our livestock, to our animals? So today we have a different option. Selenium supplements are divided in two main groups, inorganic form and organic form. In the inorganic form, we have the first generation of selenium additives, which are mainly sodium selenide or sodium selenate, are mineral source considered a product of the past because they have been authorized for as feed supplement in 1979. However, being mineral sources, they are not able to provide any selenium in the form of seleniumethanine, the safe storage form of selenium. And then we have the second big classes, which are the organic form of selenium. And those organic forms can be divided in two different groups. The second generation of selenium additive, the selenium yeast, Those forms have been authorized in the year 2000. They are produced by a biological synthesis and are basically yeast which are able to incorporate and to convert sodium selenite in selenium amino acid. When they arrived in the market, it was a big revolution for the selenium nutrition because we are the first form able to provide part of the selenium in the form of selenomethane. However, those forms they have some limits, and the limit is that on average they can just provide about 60% of the total selenium in the form of selenomethane. And on top of that, due to the manufacturing process, this selenomethane can be quite variable from one product to another, and also within the same product from one batch to another. And then since 2013, arrived in the market the so-called pure form of organic selenium. Those forms are produced by a pure chemical synthesis, and thanks to this pure chemical synthesis, those forms are able to provide 100% of the selenium in the form of selenomethane. Today in the market we have two molecules, L-selenomethane or hydroxyselenometane. So now let's have a look how to understand which source is the most bioeffective, the most bioavailable. First, let's start with the different metabolism. So when we fed animal with sodium selenite, this form is absorbed in the gut by passive transfer. However, when we fed selenomethane or hydroxyselenometane, those forms are already better absorbed in the gut. Then all the form, so sodium selenite or organic form, are all transalinated in the liver, transformed into selenocysteine and in 25-selenoprotein. However, when we fed sodium selenite, the part that is not immediately used to produce selenoprotein cannot be stored, therefore need to be immediately excreted to avoid toxicity to occur. However, when we fed hydroxyselenometane, for example, part is immediately used to maintain and to produce selenoprotein, the antioxidant enzymes, but the part that is not used immediately is not excreted, but is unspecifically incorporated into body protein, creating this body storage of selenium, this selenium insurance policy. So how to understand which source is the most bioavailable? The scientific community recognized that the bioefficacy of different selenium compounds can be assessed by considering at which extent different selenium additives are able to increase the selenium deposition in different tissues. So in a study that was published in 2013, the authors compared sodium selenite and two different selenium yeasts in broiler chicken, a different level of supplementation. And what they saw in this study is that by increasing the dose of sodium selenite they were not able to increase the selenium muscle deposition in broiler chicken. However, they saw that when they fed the two different selenium yeasts, the selenium deposition in the breast muscle significantly increased. And on top of that, they also highlighted that the two different yeasts were not equivalent in terms of bioefficacy. In fact, the selenium yeast that was providing the higher level of selenomethane was also the selenium yeast that was showing the highest selenium deposition in muscle tissues. The study more recently, a meta-analysis that integrated 13 controller randomized broiler chicken study where different sources of selenium were compared, corroborate the result that of the previous authors. In fact, those authors showed that again the mineral source of selenium are not able to increase the selenium deposition in different tissues. The selenium yeast are indeed helping to increase, thanks to this selenomethane content, the transfer of selenium into the breast muscle. However, the pure form of organic selenium, L-selenomethanin or hydroxyselenomethanin, were the form that were able to maximize this transfer of selenium, so maximize this selenium insurance policy. And more recently, a new study published this year, where again different forms of selenium were compared in broiler chicken, further confirmed that organic form of selenium are more efficient than sodium selenite, that within the organic form of selenium, the pure form, so L-selenomethanin or hydroxyselenomethanin, were more efficient than the two different selenium yeasts tested in the study, and also that the bioefficacy of selenium yeast is directly correlated to the selenium-methionine content provided by each selenium yeast. More selenium-methionine the selenium yeast is able to provide, more higher is the selenium transfer in muscle tissues. And on top of that, those authors also compare the two pure form at different doses of supplementation. And what they saw is that L-selenomethanin and hydroxyselenomethanin were exactly showing the same bioefficacy. And some years ago, another study evaluated the bioefficacy of a different form of selenium by looking at the different parameters, looking actually at the selenoprotein gene expression and selenoprotein activity. And in this broiler study, the authors fed broiler chicken with a negative control diet, so a diet not supplemented with selenium, or feeding the broiler chicken with the same diet but supplemented with either sodium selenite or selenium yeast or hydroxyselenomethanin. And what they saw is that when the broiler chicken were fed sodium selenite compared to a negative control diet, there was an upregulation of the selenoprotein gene expression and also of the selenoprotein activity. However, when the broiler chicken were fed with a selenium yeast, there was a significantly further upregulation of some other genes and as well as the activity of some selenoprotein. And then they saw that only hydroxyselenomethanin was able to express new selenoprotein genes and was the source that, compared to sodium selenite and selenium yeast, really differently modulate the gene expression of the selenoprotein as well as the selenoprotein activity, showing that this form of organic selenium is able, from one side, to maximize the selenium deposition in muscle tissues, but at the same time is also proving to be the most efficient or effective selenium source, able to differently modulate the selenoprotein gene expression and the selenoprotein activity, maximizing the antioxidant protection. So the key point of my presentation that I want to highlight is the importance to maximize the body storage of selenium. In fact, when we fed different sources of selenium, hydroxyselenomethanin, L-selenomethanin, selenium yeast, or sodium selenite, all these forms are absorbed in the gut, then transformed in the liver into hydrogen selenide, then in the novoselenocysteine, leading to the synthesis of 25 selenoprotein, providing antioxidant defense. However, when we fed, for example, hydroxyselenomethanin, the part that is not immediately used can be stored, creating this body storage of selenium. And here is the key point, because when animals are under stress, for example, heat stress is also the condition in which they are facing more challenges, more oxidative stress, and they also need more selenium and more selenoprotein. Therefore, in this stress condition, there is also higher protein catabolism, so there is some selenomethanin which is released. This selenomethanin can enter again in the bloodstream, converted in delivery into hydrogen selenite, de novo selenocysteine, and therefore increasing or maintaining the selenoprotein synthesis, maintaining the antioxidant defenses, and consequently, by creating this body storage of selenium in advance, our animals are able to maintain their productive and reproductive performance also in stressful conditions. So, in conclusion, we understood that selenium is an essential trace mineral. It occurs as selenium amino acid in the body. Selenium methane as a safe storage form of selenium that can be then converted into selenocysteine, and then selenocysteine, which is the functional amino acid present in the 25 selenoprotein. And these selenoprotein, we understood that are essential in maintaining the redox and the immune defense, and they help animals to cope with different challenges. And then, last but not the least, is that the selenium supplements are not equivalent. Organic selenium form are indeed more effective in enhancing the selenium deposition in different tissues compared to mineral form. Animals are not able to synthesize selenium methane, so we need to provide selenium in the form of selenium methane to our animals to help them to create the selenium insurance policy to help them to fight against commercially relevant stress. Within the organic form, moreover, only pure form of organic selenium, such as hydroxyseleniummethane, are showing to maximize this selenium transfer in tissues. Therefore, by measuring at which extent different forms of selenium are able to transfer selenium in tissues is a key biomarker for you to understand which source is the most effective. So selenium via selenoprotein expression is involved in the production, in the protection against oxidative stress and in the regulation of different cells function. Therefore, increased muscular reserve of selenium can enhance the resistance of our animal to stresses and diseases. And with this, I thank you very much for your attention, and I will be glad to take your questions. Thank you very much.
So thank you very much. Grazie, Dr. Marco. Very good presentation.
Thank you.
Oftentimes, we in the production, when you're in the farms, you don't really see anymore what's inside the feed. So it's very good information that we can still make improvements in how we are able to get good results by just making our nutrients even more usable to the animal, the bird. Okay, so I hope everybody has a great time listening to all our 5 speakers. And now we'd like to gather them all together for our question and answer portion. I hope everybody's here. So, hello, Dr. Marion.
Dr. Santiago is here.
Hello, everyone.
Hello.
Hello.
Very, very nice to see all of you. I know it's quite early in France and Italy. It's late afternoon in the Philippines. And very good to hear from all of you. Okay, so just for everybody's information, we have about 800 people tuning in this session today. So it's a very important session for a lot of our poultry farmers.
Yeah, amazing.
Okay, so I have we have in the question and answer part of the Zoom system, and where our participants are able to put up their questions, and I'd like to go through them one by one. Perhaps we cannot pose all the questions to everybody because we have a time to we just have enough time for this session. So I hope everybody will get one question each. So the first question goes to Dr. Corinne Morvan. Hello. How do you
Hello.
say hello in France?
Bonjour.
Oh, bonjour. I thought it was hello. Okay. So, Dr. Corinne, the question here says, there was a graph at the end that shows a competitive exclusion product used resulting to lesser medication use. Is there a study that compares brooding performance, example, livability, body weight, between flocks that use competitive exclusion products versus flocks that did not use these products?
So what I would like to say answering this question about performance, we have plenty of feedbacks from customers showing improvement of performance, talking about livability especially and growth. But what I would like to say about competitive exclusion product is that performance may not be the main target. You need to see those type of product like vaccine approach, for example. It is really a preventive health approach. Just realize that we want to play the role of the first inoculum in giving the best balanced microbiota to the bird as soon as hatch. And this is giving a day old just once for short-term production like broilers. You can have some further application for long-term production like layers or breeders. And you may know that there are plenty of things that may happen later that are going to influence the bird's performance. So see it as a preventive health approach. It is not a zootechnical feed additive that is going really to enhance FCR, for example, or things like that. But the feedbacks are really great, and all the things we see is also the improvement on foot-pod dermatitis or litter quality, and these are the type of parameters you may see above the lower incidence of pathogen colonization in the gut that you may control by doing swaps or things like that.
Okay, thank you very much, Dr. Karim. The next question goes to Dr. Marian.
Yeah.
Hello.
Hello.
Oh, bonjour.
Bonjour.
So the question here from our participant says, is it possible to improve genotypes, or I guess genetics, for both production and disease response? And I would think the question is more like, is omics now being used in genetic selection, especially against its resistance to disease pathogens like Salmonella or E. coli?
Maybe this question is addressed to the breeding companies, but I guess, and I've heard that they started to shift the selection of breeds to more trying to promote the birds with an excellent gut function and immune system. So I think it has been taken on board by breeding companies to improve the gut health and the immune system, yes. So they do not only focus on weight gain, they have a broader target now that covers the immune system, and that should lead to breeds with a higher defense, specifically to address the period of the antibiotic-free production.
All right. Thank you very much. This next question is addressed to Yago, Dr. Yago. How are you? ¿Cómo está?
Fine. Good morning, bonjour, buongiorno, guten Morgen.
The world is very small. So the question for you is, what is the recommended egg size that fits into the in ovo vaccination system?
Okay. Thanks for the question, first of all. When talking about in ovo vaccination, okay, first I have to say that there are a few things that we have to take into account. Okay, first of all is we have to provide the in ovo machines the maximum uniformity. Okay, we have to keep a good recordings of the rearing farms and the breeder stock on the first day, because we need to provide this maximum uniformity during the laying period. It will affect also the quality of Innovo vaccination. We must provide the most uniform egg size to the Innovo machine. Okay. Then another point that we have to take into account at this stage is that it will be more difficult for the young breeder stock because the eggs are also small. But the main point I want to answer regarding the question about the egg size for vaccination is that we cannot give a specific certain range of egg size, okay? Because there are few types of Innovo machines that work different one to another. So what we must know is how our Innovo machine works. Okay? Because there are few machines that are well adaptable to the egg size. So we won't have any kind of these problems with the egg size. But there are another few Innovo machines that they are not so adaptable to the egg size. So taking the recommendations from Innovo machines manufacturers, first I would try to adapt the machine as much to the egg size, and then of course try to maximize the uniformity of these eggs.
Okay. Thank you, Dr. Yago. I guess in the Philippines, or I guess in a lot of countries in Asia, when the opportunity is high, the egg sizes criteria is enlarged, and then you use a lot more of those eggs for hatching.
Yes, I would like to mention as well that, as I mentioned on the presentation, we have this database analysis service called PSOL. So if there is any kind of customer that wants to assess which is the difference regarding the egg size and also the egg hatchability or the profitability of those eggs, we have this kind of service that we can analyze with this database service that is a very specific timeline to enhance and support our customers' performance. So I would like to invite all of them who want to assess this kind of technical issue for its own profitability.
Thank you very much. This next question is for your Arlene. So Arlene, I'm not sure if this will be something that is part of your presentation, but the question says that you mentioned an increase in different immune cell counts when using beta-glucans under heat stress period. What is the main mode of action of beta-glucans in increasing immune cells?
So before I answer that question, I'd like to congratulate first our Poultry School Online this 2021. I saw it. We have around 800 attendees. Congratulations. So going back to the question, the main mode of action of beta-glucans, remember in the Philippines, not all beta-glucans are the same. Okay, so we're talking about the non-specific immune system of the animals. Once these beta-glucans was ingested or given to the animals with our animal nutrition diets, it binds, or automatically we were going to combine it to phagocytic cells or macrophages that becomes more active. So once it's activated, it produces substances, chemical substances called cytokines, which in turn causes a chain reaction and produce more phagocytic cells. So these phagocytic cells would undergo its own phagocytic action over the microorganisms and thus produce more or new macrophages. So this is the reason why when we claim that the product is really, or the mode of action, I mean, of beta-glucan, is an immune enhancer, it's very important for us to measure the surface area of the ileum, duodenum, and jejunum of the animals. And we're very fortunate through the years, more than 10 years of monitoring, it's really very important to standardize the surface area. It's not just longer the villi. The longer the villi, the better. No, it's not anymore the new schools of thought. It has to be the wider, the better. And we discovered that surface area would really give us a clear picture of the beta-glucans mode of action in the animals. I hope I was able to answer the question.
Yeah, I guess that's part of the question. The other part of the question about heat stress, but I guess it's kind of difficult to, or how to say, it takes a long period or a longer explanation to connect beta glucans with heat stress. Huh?
Actually, Doc, if I may add, in a heat stress condition here in the Philippines, we all knew very humid and very high temperature. This is really very effective because we identify it as an anti-stress strategic nutritional tool, because this will cover all the stress-related activities in the farm, not just climatic conditions, but all procedures being given to the animals. Because we're talking about beta-glucans per se. So we're talking about immune system per se. Beta-glucans, if I may add, we also discovered through our literatures, to our scientific trials, it's not effective for us to give a very concentrated form of beta-glucans, and we all knew it's available in the market because it overstimulates the immune system. So there is a specific combination of beta-glucans needed in the diet. We subjected this to interleukin-6 and interleukin-10, which I believe very important for us to monitor in terms of anti-inflammatory and anti-allergenic reactions. That's the reason why combinations or the use of a specific ratio for beta-glucans is very important. More so with the beta-1316 linkages that we used.
Okay, thank you. Good to know. So this next question is for Dr. Michelle. I put you last because I wanted you to rest a bit.
Yeah, thank you.
But you're not speaking really. But anyway,
Exactly.
the last question is, what form of selenium would you recommend to help alleviate environmental stress for broiler breeder flocks, such as heat stress condition during laying period?
Yeah, this is indeed a very good question. So basically, as I was explaining during my presentation, the key point, in particular when animals are facing different challenges, is really to help them to have these reserves of key trace mineral in the body that can be used when they are in a challenge condition. If you think that when there is animals are facing heat stress, one of the first adaptation is that they decrease the feed intake. So normally, but it's normally also the moment in which they need more nutrient, more antioxidants. So having built up in advance this selenium reserve, they are able to keep maintaining the synthesis or increase the synthesis based on their needs of those selenium protein, which are, as I explained, involved in the in maintaining the redox balance, but also involved in provide a good immune and inflammatory response. So when it comes to this, to which selenium source I would recommend, of course, due to the fact only selenium methionine can be stored in tissues, the key point when we talk about selenium nutrition is not to talk about selenium supplementation, but about selenium methionine supplementation. So we should provide the source that is able to provide the highest amount of selenium in the form of selenium methionine.
Does that mean that this kind of selenium is available not only as a feed supplementation? Is there
yeah, I mean, as I was explaining, today we have many options. We have the mineral source, which are minerals, so they are not providing any selenium in the form of selenium methionine. Then we have the selenium yeast that are indeed organic form, but they just provide around 60% of the total selenium in the form of selenium methionine. And then today we have these those pure forms of organic selenium, which are pure chemically synthesized form, and they are providing 100% of the selenium in the form of selenium methionine. So today, also based on all the studies that are present in literature, is quite clear that those pure form are those that are maximizing this storage of selenium in the body and consequently helping animals to better face all the relevant stresses that animals are facing in the field. So can be aging in breeders or layers, can be heat stress in all species, broiler layers, ruminants, and there are also some physiological stresses that are specifically related to specific physiological stages of the production. So the reproduction in breeders, the transition period in the ruminants, and in sows. Yeah.
Thank you.
Yeah, you're welcome. And thanks
I've
for the
got-
organization. Huh? Yeah.
There's still a bunch of questions here, but I'd like to, until we still have time, I'll keep on shooting the questions to you guys. The next question is for Dr. Corinne. What is the best feeding period to include in a competitive exclusion product for a competitive exclusion product in layer or broader feeding regimens? Thank you.
Thank you for this question, because it is going to allow me to give precise and detail about the application and also about the nature of the product. So you understood that competitive exclusion products are coming from commensal gut content from healthy adult birds. So it means it is bacteria that are going to colonize the intestinal tract. It is not a probiotic. I want to make the difference with probiotics. It is something different. You need to give some probiotics every day if you want them to stay within the tract. Whereas for a competitive exclusion product, those bacteria, because they are coming from chickens, they are going to be able to colonize the tract. And this is very important to understand the difference. So it is not a feed additive. We don't use competitive exclusion product in the feed. We use them by spray or by drinking water, and we make an application in a short time at the hatchery by spray, or we can also do it in the drinking water at the farm level. And from my presentation, you understood that the best period is to give it as soon as hatch. So preferably at the hatchery level, but you can also give it during the rearing of the birds or at the laying period, for example, for layers, which is the production mentioned in the question. And there for layers, we would advise above the first application at hatch or at day old at the farm level or at the hatchery, we would advise to give a further application sometime after a strong vaccination program, before transfer or just before the stress period of the peak of lay, which are the type of program we can apply for layers and also for breeders.
Yeah, this is interesting because some other guys are asking that when you are using the competitive exclusion product, is it affected by acidifiers in water?
That's also very interesting, because how it works is that it takes only a few hours for those bacteria content, gut content, to colonize the intestine of the birds. So it means after 5-6 hours, we can consider that the microflora has established itself. It means whatever you're going to do later shouldn't affect the establishment of this microflora, except antibiotics, I mean. But normal practices that you would have at farms level, like using acidifiers, for example, is not going to alter the efficacy of such a product.
Thank you. Good to know. The next question is for Dr. Marian. So here the question is, what is the interrelationship between nutrition and immunity, especially those with regards to poultry gut health? Does genes or genetics have anything to do as well in governing gut health and immunity in poultry? I can't hear you.
If I understand correctly, the question is about relationship between nutrition and immune system, correct?
Yes, and how all these genomics that we've studied are being used to improve the condition situation in our, especially when we are selecting our birds to be growing very fast, and they're growing the time, especially in the Philippines when we are just growing broilers, for only for 4 weeks. Then this is quite important to know.
Yeah, by feeding the birds, you not only feed the animal, you also feed the intestinal gut microbiota, and we know more and more now how it's the connection between the microbial population, how they ferment and digest the nutrients that are not absorbed by the birds, and the secretome. I mean, all the metabolites that are released by the intestinal gut microflora directly interact with the human cells. So everything is connected, and this is now also translate into concepts like the nutribiosis that we are promoting, that we need to understand every part of the system to have this what is covered by the concept of holomix as well. So we have to understand everything from the nutrition, from the immune system, from the microflora, together with the environmental factors to maximize the potential of the nutrition and the breed. So by understanding how everything is interconnected, we can, in the future, provide to the production system the best breed with the best diet, and reducing the use of medication. But we need to know exactly how everything is interlinked. And we think now that we have the tools to better understand.
Okay. Yeah, from the presentation, a lot of information. A lot of our people, I guess, a lot of participants have, find it quite interesting. A lot of information that are not really known to a lot of people in the industry. So keep it up. Thank you very much.
If I may, I would like to add that what is missing for now is to better understand how it is consistent across the different models and across the different environmental factors that differ from one side to another. So by feeding more and more example on the influence of the different climate, the different production practices, the effect of the different diet, we will continue to building this library of data and provide the most accurate practice according to the environmental conditions across the global market.
Yes, I agree. It's like, I guess a lot of your studies have been done in a European setting or European microenvironment, and it's quite, I'm sure it's going to be different in other parts of the world, especially in Asia.
For sure.
Yeah. Okay, the next question is for Dr. Santiago de Verges. So our participant is asking, what causes the eggs to spoil too early?
Okay, thank you, Dr. Elias. Well, when talking about early death, mortality, and incubation, there are a few things that we have to take into account. Okay, that if we put together, they will give us better or worse results. Okay, but we have to analyze what can cause this kind of early chick mortality in incubation. Okay, especially I have never been in Philippines. I hopefully we could meet soon in Philippines. But as far as I know, there are high temperatures, high humidity as well. So we have to, the first point we should take into account is the kind of storage and the temperature and the length of the storage of the egg. So first point, once the egg is laid, try to collect them as soon as possible, because as soon as the egg is already laid, the embryo starts increasing the number of cells that are on the yolk. So we have to stop this kind of reproduction of the cells. So we have to collect that, all of them, at least my recommendation 4 times in a day, okay, and try to store them as soon as possible. Then also the temperatures, in my opinion, from my point of view and my experience, would be no higher than 18 degrees. Try to keep these eggs under 18 Celsius degrees. And once we have these eggs already stored, another thing that I would recommend is to take special focus is on the length of the storage. Okay, we all know that now with the COVID situation, almost everyone is storing these eggs for a longer period. So we try to give these eggs the best conditions for keep these cells stopped. Okay, we want to start the reproducing of the cells once we have these eggs already incubating. We have to keep these eggs that are already stored under the best temperature conditions. Okay, try to keep them as possible under 80 degrees. And then if we have a longer periods of storing, what I would recommend that also genetics companies recommend is to provide them a short period of incubation, long storage tanks, okay, before putting them on the incubation tank. We have seen that there are very good results when we store for a long period these eggs. And in my opinion, it works really well to provide this short period of incubation with long lasting eggs. Okay, so these are the main things that I would focus when talking about the early death mortality in embryos, that are the length of the storage and also the temperature. These are the 2 key points, in my opinion.
Okay. Although I'm quite sorry, Dr. Yago, your audio doesn't come through very well. Perhaps something wrong
sorry,
with the
I'm a little bit
microphone.
far from the computer.
Yeah. And anyway, I'm quite sorry. Messi already went to Paris. So congratulations, Corinne and Marian. Congratulations to you. So the next question is for Arlene. Our participant is asking, how can beta-glucan stimulate macrophages? What is the difference between activated and non-activated macrophages in terms of function?
Okay, thank you very much, Doc, for that question. Macrophages per se can be activated or classically considered as an activated macrophages when it is being stimulated by our what we call interferons, lipopolysaccharides, or the LPS, in order for it to release the nitric oxide needed. Now, once it's being activated or stimulated, this is all very necessary for intracellular pathogens to be well taken care of. So once it has been activated, the stimulation of the interleukin 4 or interleukin 3 will be there to produce interleukin 10 that would help transform growth for the immune system per se. So we call it activated macrophages once beta-glucan was ingested, and it releases the chemical substances called cytokines per se. So these cytokines will lead us to build more immune cells. On the other hand, the inactivated, I would say, or the non-specific immune cells is coming from the first line of defense in the immune system. So what are those? These are the macrophage per se, the neutrophils, or the dendritic cells, or the first line of defense, which are indeed very important for the first function against infection or injury. In the Philippines, we all knew what's the main function of the beta-glucans, what's the 3 major pathogens that we handle. Here, under this kind of temperature, we have to monitor the E. coli presence, the salmonella, and of course, the Clostridium perfringens. In return, in order for us to quantify the efficacy based on our research, we have to identify the surface area being given by the animals. So these are all has to be provided with numbers, with scientific proof. I hope I was able to answer the question, the difference between activated and inactivated macrophages.
Thank you. Thank you, Arlene. In connection with that information, for Dr. Corinne, there's a participant is asking, can salmonella vaccines, I guess the live vaccines, can they be removed if the competitive exclusion products will be used?
This is a decision to make depending on the pressure you may have on your farms, and at least I have some data showing, and there are some published data showing that Avigard is very effective, giving some protections against salmonella, and at least those type of product, competitive exclusion product, have been developed first to face salmonella issues on farms. At least there is, I'm thinking of one publication showing some very interesting effect of those type of product in comparison with live salmonella vaccine against Salmonella enteritidis, for example. In this publication, the authors even show that there was a synergistic effect and the protection was as good as the vaccine one. I think it's you need to have a cost point of view, and something you need to have in mind is that when you are using a salmonella vaccine, you are targeting one strain or two strains now. When you are using a competitive exclusion product, you are the purpose of that is all salmonella. All salmonella strains are going to be reduced by using a competitive exclusion product. So it's not quite the same approach. So you need to adapt it to your context. And the cost is not the same also.
Right. Yeah, but currently in the Philippines, government is trying to push for salmonella-free flocks. So it's a huge step for a lot of farmers.
If I may just add something about salmonella live vaccine with those type of competitive exclusion product, is that you may not use the competitive exclusion product before the salmonella live vaccine because it is so effective against salmonella that is going to prevent the efficacy of the vaccine. So you need to use it together or after.
Okay, thank you very much. We still have time. So I have a question for Dr. Santiago. For Dr. Perez. So with regards to you've been monitoring vaccinations at the hatchery, have you made some difference, or have you seen some difference in hatchery vaccinated birds in a chick room where it's tunnel ventilated as against a chick room that has the HVAC ventilation system? Because in Asia, I'm sorry, because in Asia, or at least in Southeast Asia, about 95% uses tunnel ventilation in the chick room.
Well, in my opinion, first of all, thanks for the question. When we prepare the vaccines, it is always recommendable to have a kind of ambient environment as clean as possible. Okay, so if we are preparing the vaccines in this kind of rooms, it is totally, for sure, recommendable to have a good ventilation. Okay, we know that once we open the vaccines, sometimes there is more particles that remain on the air. So it is totally recommendable to have good ventilation in the vaccine room. Okay, for sure. So totally agree to have recommendable for sure to have good ventilation in preparing the vaccine to provide the most sterile kind of vaccine solution to our birds, okay, and try to not contaminate it with other kind of pathogens that could be present on the air or other kind of environment.
All right. I guess it would be interesting to see what's the difference.
Well, do you mean if we have data regarding the difference in the vaccine application regarding the ventilation on the room or not?
Right.
But as far as I know, at least as far as I know, I don't have that data. But for sure, we have to prepare vaccines. We want to prepare them. The the maximum quality. Try to avoid any kind of contamination. So if the ventilation helps to reduce this kind of contaminations, it is totally recommendable from my part to ventilate from the best way the rooms where we prepare the vaccine.
Okay. All right. So we still have some more questions here. This is for Dr. Corinne. By the way, for information, Dr. Corinne has the most number of questions from our participants. Okay, that's good. I just would like you to know. So the next question is for Dr. Corinne. Competitive exclusion is one of the good measures to combat pathogens, but because of high production costs, do you have some cost analysis data to justify the inclusion of these products in our production systems?
That's a good one, because this is the basics of all the decisions we make in our poultry industry about the costs and the feedback. I would just come back to the first answer, or one of the first questions I had about how we should look at those type of products. And when we are talking about vaccines or protection against salmonella, of course you are going to apply a cost per chick on the vaccine, which is exactly the same you're going to do when you apply those type of product, competitive exclusion product. You will think of it as a cost per chick, not a cost per feed or something like that. And those type of application is much more cheaper than having a product added in the feed as a feed additive that you would use during all the life of the bird. So this is a calculation you need to make in your own economical context. Then depending on the result, the situation, we don't have trials data on that. We have plenty of feedbacks from customers where we can show we have some positive incidents by using Avigard, especially because the effect on mortality is very, very important. And you know that mortality is a big parameter when you analyze mid-cost productions. That's the same when you look at FCR, because of course, if you improve the microbiota, you expect to have better nutrients utilization. But again, this is not the main purpose of the product. The product is really targeting pathogen control, Salmonella, E. coli, Clostridium perfringens. This is really the target of the product. So as soon as you have a better health, you would expect better performance. So all calculations and economical approaches need to be adapted to local context. And for sure in Asia, it will be different from what we have in Europe or in US, for example.
Thank you very much. We come to the end of our Q&A because we don't have the time anymore. I'd like to thank first our participants for their questions. I hope you guys enjoyed all the questions. And then I'd like to thank you, the speakers, for gamely answering all these different questions.